everyone, welcome to PW NEET English YouTube channel. I'm Shalini Ma'am, your botany teacher, and in this lecture we are going to explore the chapter molecular basis of inheritance, which I'm sure a lot of you are petrified of. Before we start, let me tell you one thing. Push away all the fear you have around this chapter. Let's just look at this chapter with a fresh perspective. Let's try to understand this with the love for biology. And the main hero of this chapter is DNA. You will learn about the structure of DNA, how it functions so beautifully, and by the end of this chapter, I promise you that you would have fallen in love with DNA. And this does carry high weightage, so I will be sharing class notes along with DPP questions for you to practice on a free batch which you can access on the PW app. The batch is called Complete Botany by Shalini Ma'am. You can access that using the link given in the description below. Now, without any further delay, let's get started. So let's begin by talking about DNA, which, like I mentioned earlier, is the hero of this chapter, right? So we'll start off by understanding the structure of DNA, which of course you would have covered in a chapter called biomolecules, but we'll just go through this one more time. DNA, similar to your polypeptides and polysaccharides, is a polymer. In fact, it's a very long polymer. What do we mean by the term polymer here? Poly means many. So in a polymer, there are many similar units that keep on repeating and give rise to a long chain-like structure. Okay. So DNA also is a polymer. That means it has many repeating units, one after another, that are interconnected by means of chemical bonds. Okay. What kind of a polymer is DNA? What are these beads made up of? Is it pearl? Is it a red colored bead? Is it a coral? What is it made up of? The beads or the individual units in DNA is made up of what we call deoxyribonucleotides. Okay, so each individual repeating unit here is called a deoxyribonucleotide, and a long chain of that is called a polynucleotide. And within the DNA molecule, you have two such polynucleotide chains in most cases. Okay, in some you might have just one chain, and most of them, a vast majority of them, you will have two such chains. All right. So, how long this polynucleotide chain will be varies from one species to another. In fact, for every species, the length of the DNA is a characteristic feature. Like, for example, when you consider humans that are normal, humans that have 46 chromosomes, let's say, if you unwind the DNA from the chromosome, for all the 46 chromosomes and, attach them end to end and look at its length, the length of my DNA is going to be equal to the length of every normal human's DNA. And how do we measure the length here? Unlike how you do it in math, where you use terms like centimeter, millimeter, and kilometer, here we use numbers. We're basically measuring the length of DNA using how many nucleotides are present in that long chain. Okay, so the number of nucleotides is the unit that we use to measure the length of DNA. There are 4 examples given in your NCERT textbooks which you must memorize. Of the 4 examples, 2 are of viruses, 1 is of a bacterium, and 1 is humans. Okay, so let's take a look at that. First, let's look at the viruses. Viruses being very simple organisms. Can we even call them organisms? Whatever. They have shorter DNA. Okay, so there is one virus that infects bacteria. It's called a bacteriophage. That is called bacteriophage 5X174. In this particular virus, DNA has only one chain. If it has only one chain, we call it single-stranded. Strand is like chain, okay? So if it is single-stranded, obviously we will measure the length using how many nucleotides are there. Since this this bacteriophage has single-stranded DNA, the length of its DNA is about 5,386 nucleotides. Moving on, we have another bacteriophage called bacteriophage lambda, which has double-stranded DNA as its genetic material. When I say double-stranded DNA, I mean to say that there is one DNA chain like that, one polynucleotide chain like that, and another one. Double means 2, right? And there will be pairing between these two. This pairing is called base pairing. More about why we call it base pairs and all of that as we move on in this chapter, you will understand. So if at all the DNA is double-stranded, we will measure its length in terms of base pairs. This is one base pair, this is the second base pair, this is the third base pair, and so on. Okay, in one base pair, there will be 2 nucleotides involved. Alright, so in bacteriophage lambda, you have 48,502 base pairs present. Then we have a bacterium called E. coli. Okay, so in E. coli, we have double-stranded DNA as the genetic material, and its length is 4.6 into 10 to the power of 6 base pairs. And finally, we have a eukaryote, a human DNA, one set haploid content, that is 3.3 into 10 to the power of 9 base pairs. Even this is double-stranded. These numbers are important for you to remember. There will be questions asked based on this, maybe in the form of match the following or something. So make sure you have memorized all of these values. Okay. Now we'll move on. I mentioned how DNA is a long chain of repeating units, right? The individual unit is called deoxyribonucleotide in case of DNA. RNA also is a nucleic acid. In RNA, we call it ribonucleotide. Okay. So in general, in nucleic acids, the monomer is called a mononucleotide or simply a nucleotide, and the long chain is called polynucleotide. Okay. Now, there are 4 different types of nucleotides that you can find in DNA and RNA. So here I've represented them in 4 different colors just to help you understand that there are 4 different types. How are they different from each other? We learn as we move on. It's not like within DNA there's purple color ball and there's an orange color ball, nothing like that. This is only for visual representation of how nucleotides are of 4 different types. Now, when these nucleotides form a chain, obviously when you're stringing together beads for your necklace or something, you use thread, or you might use some metals like gold, silver, whatever. But in our body, when molecules have to form polymers, then there will be chemical bonds formed between the monomeric units. Over here, in order to form the long chain, the bond that forms between nucleotides are called phosphodiester bonds. Why it's called so, we'll learn later. For now, just remember that. Now we look at the structure of one nucleotide to start with. Each nucleotide, one single bead, okay? So each nucleotide is made up of 3 distinct components. Yeah. What are the 3 components? At the center of the molecule, you have a sugar, a monosaccharide. Attached to the sugar, you have a nitrogenous base and a phosphate group. So you think of this as the sugar molecule. On one hand, it is holding on to a nitrogenous base. On the other side, it's holding on to a phosphate group. The sugar, along with the nitrogenous base and the phosphate group, is called a nucleotide. It's not going to be spherical, globular like this. This is only for representation. What it actually looks like is kind of like this. Again, this is not like accurate, just for your understanding. Okay. So the sugar is at the center. One side it is attached to phosphate, on the other side it is attached to a nitrogenous base. Okay, now we'll understand the structure of sugar, phosphate and base in detail. Here we'll start with sugar. The sugar is a monosaccharide. It is made up of 5 carbons. It has a ring-like structure. Since it has 5 carbons, we call it a pentose sugar. Penta means 5. So it is a 5 carbon monosaccharide. And like I mentioned earlier, in a nucleotide, sugar is the central molecule. There are 2 different types of sugar, though both RNA and DNA are nucleic acids. Structurally, there are a few differences between the two. So the sugar that you find in RNA is called ribose, and the sugar that you find in DNA is called deoxyribose. There's not too much of a difference between the two. I'll tell you, there's just one difference. It's very easy to make out. Here, we'll start off with ribose. Ribose has, I told you, pentose sugar, 5 carbons. So does deoxyribose. We'll start numbering the carbons in clockwise direction starting from here. Here you have 1, 2, 3, 4, 4 carbons. Clockwise, we'll number them 1 prime, 2 prime, 3 prime and 4 prime. The 5th carbon is outside the ring. Let's call this 5 prime. Okay. Look at the structure of deoxyribose. It's pretty much the same. There are 5 carbons here. There's 1 here, 2 here, 3 here, 4 here and 5 here. Okay. So the structure is the same. To the first prime carbon, you can see in both cases there is a hydroxyl group and a hydrogen attached. To the second carbon, in ribose, you have a hydrogen and a hydroxyl. There is a 2 prime free hydroxyl group here. As opposed to that, if you look at deoxyribose, specifically at the second carbon, instead of a hydroxyl group, what they have here is hydrogen. They don't have a free hydroxyl group here. Where? Specifically at the second prime carbon. That is why we call it deoxy, because there's one oxygen less here at the 2 prime carbon. That's why it's called deoxyribose. Otherwise, if you look at every other carbon, it's the same in both cases. The difference only is in the 2 prime carbon. That's why we call this 2 prime deoxyribose. Okay. So at the 3 prime end, you have a free hydroxyl group in both cases. Carbon here has hydrogen, carbon here has hydrogen, 2 hydrogen and 1 hydroxyl group at the 5th carbon. All of the other things remain the same. The difference only lies in the 2 prime carbon. Okay, so that's the difference between ribose and deoxyribose. Next, let's talk about the second component found in a nucleotide, which is called a nitrogenous base. Nitrogenous bases, of course, as the name indicates, has nitrogen, but along with nitrogen, the nitrogenous bases also are composed of carbon, hydrogen and oxygen. Now, I'll tell you the reason why we number carbons in sugar as 1 prime, 2 prime, 3 prime and not 1, 2, 3. If you look at the nucleotide, a nucleotide has sugar, it has a nitrogenous base too. Sugar also has carbon, nitrogenous base also has carbon. Now, if I were to say the first carbon of this nucleotide, would you think about the carbon of the sugar or the carbon of the nitrogenous base? You would be confused, right? So, in order to avoid such confusions, what we do is while we're numbering carbons in the nitrogenous bases, we go 1, 2, 3, we number it like that. And when you're numbering carbon in sugar, you go 1 prime, 2 prime, 3 prime. Some people call it 1 dash, 2 dash. It's fine, anything. You can call it anything. But I would prefer to call it 1 prime, 2 prime, 3 prime. Okay. Yeah. So this is the nitrogenous base. Nitrogenous bases are of two types. You have purines and you have pyrimidines. Both of these are heterocyclic rings. If you look at their structures, purines have a double ring structure, 1 and 2, 1 and 2. Pyrimidines have single rings. Okay. So purines, the name is small, but their structure is larger. This has 2 rings. And pyrimidines, the name is relatively longer, but the structure is smaller. It has 1 ring. There are 2 different types of purines and 3 different types of pyrimidines. Which are the purines? Purines are adenine and guanine. They are represented in short as A and G. Moving forward, every time I say A, G, C, T, U, you should understand that I'm talking about nitrogenous bases. Okay. And then pyrimidines are of 3 types. You have cytosine, thymine, uracil. Okay. Now, are all of these nitrogenous bases found in both DNA and RNA? Not really. Some of them are found in both. Some of them are exclusive either to RNA or to DNA. Adenine, guanine. The purines can be found both in DNA and RNA. Okay, so adenine, guanine along with cytosine can be found in DNA as well as RNA. Whereas thymine is found only in DNA. Uracil is found only in RNA. Okay, so DNA has 4 different types of nitrogenous bases: A, G, T, C. RNA has A, G, T, U. These 3 are common to both. T is found only in DNA, U is found only in RNA. Okay, now let's speak of how the bond formation happens between a sugar and a nitrogenous base. To the first prime carbon of sugar, let's consider ribose for now. In ribose, this is the first prime carbon, right? So, to the first prime carbon, the nitrogenous base will get attached. And this happens by means of what we call a glycosidic bond or a glycosidic linkage. Now, when the sugar is attached to the nitrogenous base, this structure that forms as a result, sugar plus nitrogenous base, this structure is what we call a nucleoside. Has a sugar and a base, that is nucleoside. Okay. We give nucleoside different names based on which sugar it has and which nitrogenous base it is attached to. assuming the sugar that we have here is ribose, you can say that when ribose is attached to adenine, we call the nucleoside adenosine. You would have heard about adenosine triphosphate, right? Adenosine in ATP, it is nothing but a nucleotide. Okay, so ATP is a nucleotide. So adenosine comes from here. Adenine, if it is the nitrogenous base attached to ribose, then the nucleoside is adenosine. If it's guanine, it is guanosine. If it is cytosine, it is cytidine. In case of RNA, there will be no T. So if it is uracil, it is not urine but uridine. Okay. So you can see the structure of how these nucleosides look. There is a 2' hydroxyl group which makes you, which helps you understand that it's a ribose sugar. And then you have these nitrogenous bases attached to the first prime carbon in every case. Okay. Now, if only the sugar was deoxyribose, then what would you call these? If the sugar was deoxyribose, then there's only one change. There is a prefix added to the name of the nucleoside. Instead of calling it adenosine, we call it deoxyadenosine. Okay, so if the sugar is deoxyribose and the nitrogenous base is adenine, the nucleoside is called deoxyadenosine, deoxyguanosine, deoxycytidine. There's no U in DNA, so it is deoxythymidine. Okay, so that's about the nucleoside. Moving on, let's speak of the phosphate group. The phosphate group, you know, H3PO4, from that you derive from phosphoric acid, the phosphate group. It is negatively charged. How does this phosphate group attach to the sugar? It attaches to the sugar specifically at the 5' end, or I'm sorry, not the 5' end, to the 5' carbon. Here you know there is first prime carbon, second prime carbon, third prime carbon, fourth prime carbon, fifth prime carbon. This is shown for deoxyribose, but is the same for ribose too. So this 5' carbon has 2 hydrogen, 1 OH, CH2OH, right? So to this 5' carbon, whichever OH group is attached, this one specifically, this one, this reacts with the OH group of the phosphate. Phosphate has H3PO4, right? So it has a hydroxyl group here. So the hydroxyl group of the phosphate reacts with the hydroxyl group attached to the fifth prime carbon of the sugar. A molecule of water is lost, condensation reaction, and a bond forms as a result. And that is how the phosphate gets attached to the 5' carbon of the sugar. And the bond that forms as a result is what we refer to as the phosphoester bond. Do not get confused between the phosphoester bond and phosphodiester bond. I told you earlier that when nucleotides are joined to each other to form the long chain, we call the bonds that link these nucleotides together a phosphodiester bond. But over here, it is not a phosphodiester bond, it is a phosphoester bond because there's only one ester linkage here, right? So that's why this is called phosphoester bond. Now, if you have the sugar, the phosphate and the base together, what we call that complete monomeric unit is a nucleotide. Okay, so nucleotides also have their own names based on whether the sugar is ribose or deoxyribose, based on which is the nitrogenous base that they are attached to. Assuming it is a ribose sugar, if the nitrogenous base is adenine, you call it adenylic acid. If it is guanine, you call it guanylic acid. If it is cytosine, you call it cytidyllic acid. If it is uracil, then you call it uridylic acid. This is for ribose, again no T here. If it is deoxyribose, then you call it deoxy, this has to be deoxy, my bad, deoxyribose. Then you add the prefix deoxyadenylic acid, deoxyguanylic acid, deoxycytidyllic acid, deoxythymidyllic acid, there's no U here. Okay, so this is how you call, you name nucleotides, okay, DNA nucleotides and RNA nucleotides. Now that we've learned about how a single nucleotide comes together, all of the components in a nucleotide comes together, we'll try and understand how a long chain of nucleotides forms. Okay, for a long chain of nucleotides to form, bonds have to form between two nucleotides. Okay, so here we have nucleotide 1 right here, and here we have the second nucleotide represented in orange color. So this is a regular nucleotide that has a nitrogenous base attached to the first carbon. Doesn't matter if it's ribose, deoxyribose. Let's just understand how two nucleotides are linked together. So here you have the nitrogenous base, here you have the sugar, here you have the phosphate group. This is one nucleotide here, and here you have another nucleotide, right? So there are two nucleotides here. In between these two nucleotides, you can think of them as beads, like I mentioned earlier. This is one bead, and here you have another bead. There should be a link formed between these two, right? So how does this link form? By means of a chemical bond. How that bond forms is what I'm trying to explain to you right now. We are looking at the formation of a phosphodiester bond. It's very similar to how a phosphoester bond formed earlier. But here the bond formation is happening between two different nucleotides. In the formation of phosphodiester bond, there are two molecules of, two nucleotides involved. Of one nucleotide, the 5' carbon is involved. Okay, so this is the nucleotide. Let's consider this as nucleotide 1. In this nucleotide, the phosphate group attached to the 5' carbon is involved. In the other nucleotide, the hydroxyl group attached to the 3' carbon is involved. Okay. So here in this first nucleotide, phosphate group is taking part. In the second nucleotide, 3' hydroxyl group OH is taking part. We're not talking about 2' hydroxyl. Don't get confused. Where is OH coming from? In DNA it is not there. That is about 2'. Here we are talking about 3' in both ribose and deoxyribose. 3' has a free hydroxyl group. Okay. So the hydroxyl group present in this phosphate and the hydroxyl group attached to the 3' carbon, between both of them a reaction happens, a molecule of water is lost, again condensation reaction happens, and as a result you can see the formation of phospho 1,2 diester bond formation between these two nucleotides. This is called a phosphodiester bond. Here you have phosphoester bond because this is not attached to another nucleotide. So between the sugar and the phosphate, what you have here, this single bond, that is a phosphoester bond. Here on either sides of the phosphate, you have two ester linkages. That's why we call this phosphodiester bond. So similarly, at the 3' end of hydroxyl group, to the 3' end of hydroxyl group here, oops, I chose the wrong pen. Okay, so you have a hydroxyl group here. To this hydroxyl group, another nucleotide will come and attach by means of its phosphate group attached to its 5' end, and further more and more beads will get added to this, more and more nucleotides will get added to it, and the chain will become elongated. Okay, always remember that newer nucleotides will come and attach only at the hydroxyl group here. Free hydroxyl group at the 3' end, newer nucleotides will not go and attach to the 5' end. So the chain essentially elongates from the 5' direction to 3' direction. Okay, so this is the phosphodiester bond formation. Now when a long chain forms, such a long chain forms, which color should I use? Yellow maybe. You know, attached to this carbon right here, attached to this carbon, to the first carbon here, there is a free phosphate group, and this phosphate group is attached to the 5' carbon. So at this end, you have a free phosphate group which is attached to the 5' carbon. On the other end, you do not have a free phosphate group, but you have a free hydroxyl group that is attached to the 3' carbon. So if you look at the chain, it is polar. It has polarity. On one end, there is a free phosphate group. On the other end, there is a free hydroxyl group. Since the phosphate group is attached to the 5' carbon, you call this end the 5' end. Since this hydroxyl group is attached to the 3' carbon of this nucleotide, you call this the 3' end of this chain. This chain has polarity. On one end, you have a free phosphate group. On the other end, you have a free hydroxyl group. Whichever end has the phosphate group, that is the 5' end. Whichever end has the free hydroxyl group, that is your 3' end. Okay. Now, we've learnt about what is a sugar, a phosphate, a base, a nucleoside, nucleotide, phosphodiester bond formation. There is one point of confusion that students face. What's the difference between adenine, adenosine, adenylic acid? Let's clear that confusion. This is adenine is just a nitrogenous base. Add this nitrogenous base to a sugar, it becomes a nucleoside, it becomes adenosine. To this nucleoside, add a phosphate, it becomes adenyllic acid. Okay. Nucleotide minus phosphate becomes nucleoside, adenosine. Nucleoside minus sugar, just the nitrogenous base, adenine. Okay. Sugar only, ribose or deoxyribose. Nitrogenous bases, purines, pyrimidines, they are different types. Phosphate group is common to both RNA and DNA. The only thing that does not change between DNA and RNA is the phosphate group. Okay, so keep that in mind. Now, let's talk about how we got to know about the structure of DNA. Of course, today when people talk about DNA, you see DNA around everywhere. Like even in toothpaste ads, Dettol ads, everywhere behind there is like DNA spinning around. You know that its structure is very fancy, cute looking molecule. It is like a staircase that is twisted, like, or a ladder that is twisted. So basically we know about its structure today, but back in the day people did not know about the structure of DNA, and a lot of people did a lot of research trying to find out and elucidate the structure of DNA. Today we're going to learn about some of those scientists and researchers' contributions and who have helped us understand the structure of DNA. So we'll start off with the work of Friedrich Mischer. So he's the first person to have identified the substance inside the nucleus. He found out that it was acidic, okay, and he called it nuclein. This was in the year 1869. He identified DNA as an acidic substance that was present inside the nucleus, but he didn't call it DNA, he called it nuclein. Later, many years later, two people, Rosalind Franklin and Maurice Wilkins, both of who were working in the same lab, they performed X-ray diffraction studies on DNA. So they basically isolate DNA from cells, they crystallize it and they pass X-rays through it. Obviously, X-rays will not pass through like a straight line, the X-ray will diffract. You would have learnt about this in physics. And based on the diffraction pattern of X-rays, they kind of try to understand how the structure of DNA could be at the molecular level. So basically, they figured that DNA has two chains. They figured that DNA has, it is coiled, the two chains are coiled. And they also figured that the width of DNA is uniform throughout. Okay, so these were some of the contributions of Rosalind Franklin and Maurice Wilkins. They found out about this through their X-ray diffraction studies. Furthermore, there was another scientist called Erwin Chargaff. Okay, so what he did was he analyzed DNA chemically, specifically double-stranded DNA. Every time he analyzed DNA and found out, okay, how many adenine is there here, how many guanine molecules are there, how many cytosine, how many thymine, he repeatedly got results where the amount of purines was always equal to the amount of pyrimidines. Purines, how many ever purines were present, whatever was the concentration of purines, the same amount of pyrimidines were present in every double-stranded DNA sample that he analyzed. Okay, which means Ag is equal to Ct. Also, what he noticed was the amount of A was always equal to the amount of T, the amount of G was always equal to C. Okay, so this ratio between AT and GC was always constant and equal to 1. This was his contribution. Based on Erwin Chargaff's results, based on Maurice Wilkins and Rosalind Franklin's observations, two people, James Watson and Francis Crick, they proposed what is today called as the double helical model of DNA. Okay, so Watson and Crick were even awarded the Nobel Prize for the same along with Maurice Wilkins. Rosalind Franklin had passed away by then. So the three of them were awarded the Nobel Prize. So basically in their double helical model of DNA, they suggested that there is some sort of base pairing happening between the two strands of DNA. Already Rosalind Franklin and Maurice Wilkins had told that there are two strands. Already they had told that it is helical. And Erwin Chargaff had already told that A is equal to T, G is equal to C. They kind of put together all the dots, they connected all the dots and they kind of suggested this whole base pairing theory. Not theory, yeah, they suggested that. Okay? Yeah. So at the beginning of this unit, you have information about scientists, Watson and Crick. information about scientists is also important. This should have been done along with principles of inheritance and variation, because that is the first chapter in the unit genetics. But this is more relevant here, so I've added that here. Let's learn and understand a little bit about the lives of uh, James Watson and Francis Crick. So these are the two guys. Let's see what NCERT wants us to know. Yeah, we'll start off with James Watson. James Dewey Watson. He was born in Chicago on the 6th of April in 1928. In the year 1947, he received his BSc degree in zoology. During these years, he was very, very interested in bird watching. He was a birder basically. And his interest from bird watching and interest in birds, it kind of, transitioned into interest in genetics. And it was made possible for him to learn and understand genetics better when he received a fellowship from Indiana University, which is in Bloomington. Over there, he received his PhD in the year 1950, and he basically studied the effect of X-rays on viruses called bacteriophage multiplication. Okay. So, James Watson met Crick, and both of them were working on trying to understand the DNA structure, molecular level, how is the structure, how are the two strands linked to each other, attached to each other, what are the kind of bonds that are found there, what is its length and all of that. They were interested, they had mutual interest in that field. So they started to collaborate in their research. So initially when they started off, whatever they attempted, it was not very successful. But eventually later in their second attempt, based on more experimental evidence, of course I told you where they got this evidence from, and better appreciation of the nucleic acid literature, they proposed the complementary double helical model of DNA. So speaking of Crick, Crick was born on the 8th of June in 1916 in Northampton, England. He studied physics. He studied physics at the University College of London and obtained a BSc in the year 1937. He also completed his PhD in 1954, and his studies also, like I mentioned earlier, was on X-ray diffraction of polypeptides and proteins. Basically, they perform X-ray diffraction to find out about the molecular structure. Okay, if they want to elucidate the structure of any molecule, X-ray crystallography is something that's commonly used. Okay, so a critical influence in Crick's career was his friendship with J.D. Watson. The two of them, they were friends. Then a young man of 23, he was leading in 1953 to the proposal of the double helical structure for DNA in the replication scheme. Crick was made an FRS in the year 1959. Okay, so the two of them have received many, many awards together, including the John Collins Warren Prize and the Lasker Award, the Research Corporation Prize, and the Nobel Prize too. Okay, so all of these awards have been, honored to both Watson and Crick. Okay, so now we'll move on and learn about the salient features of DNA according to the double helical model of DNA proposed by Watson and Crick. So we already know that DNA is made up of polynucleotide chains. The reason it's called double helical model is because it has two strands. So it is made up of two polynucleotide chains. You can see over here, this is one polynucleotide chain. Let me draw it instead. This is one polynucleotide chain, and this is the other. You have two chains here. Okay, so this is one and this is the other. It is made up of two chains. You can see how there are individual nucleotides here. This nucleotide has a sugar, a base, and a phosphate group. This is one nucleotide. This is a sugar, base, and phosphate group. This is a second nucleotide. There is phosphodiester bond formed between the two nucleotides. All of that you can see in both cases, right? So if you look at the structure of the uh, two chains here, you can make out how it looks like a ladder, right? So in a ladder, how you have backbones and then you have the steps or the rungs, even over here, you have backbones, right? Have backbones and you have rungs or the steps, right? What is the backbone made of? The backbone is made of alternating units of sugar and phosphate. You have phosphate and sugar and phosphate and sugar and phosphate and sugar alternating with each other. In both cases, here also you have sugar, phosphate, sugar, phosphate, sugar, phosphate, right? And then what is projecting inside are the nitrogenous bases. Here you have nitrogenous base from this strand, here you have the nitrogenous base from this strand. They're both facing each other. Okay, so this is one nitrogenous base, this is one nitrogenous base, so on. For every nucleotide, you have the nitrogenous bases facing inside. So the backbone is made up of sugar and phosphate alternating and the bases project inside. Like I mentioned earlier, if there is a free hydroxyl group, that becomes the 3' end of the DNA strand, and wherever there is a free phosphate group, that becomes the 5' end. So it is polar. It has polarity. And we say that the double-stranded DNA is anti-parallel in nature. It has anti-parallel polarity. What do we mean? We mean to say the two strands run parallel to each other, but the polarity are reversed. If there is 5' end at this end of this DNA strand, of the other DNA strand at the same end, you will not have 5' end, instead you will have 3' end. If there is 3' end here, you will have 5' end. Why is that so? Why can't both be 5'? Why can't both be 3'? If only this was also 5'. Forget about that strand. Let's say this is one strand. We'll have another strand here that has 5' on this end, which means the phosphate group is here. It is attached to the sugar and it is attached to the nitrogenous base here. That is how two 5' come together. If this was the case, then the nitrogenous base of this strand will not face the nitrogenous base of the other strand. For base pairing to happen between nitrogenous bases, the nucleotides of the other strand have to be ultafied. It's only then that the two nitrogenous bases can face each other and form hydrogen bonds for stability, right? So if it is like this, obviously you cannot expect base pairing to happen, which makes it unstable. As a result, one of the strands you can find 5' to 3'. On the other strand, you can find 3' to 5'. This brings the nitrogenous bases opposite each other. Okay, so that is anti-parallel nature or anti-parallel polarity. This is 5' to 3', that is 3' to 5'. What do we have next? I told you that the bases are opposite to each other. There is base pairing that happens between the two nitrogenous bases present in the two DNA strands. So in case there is a purine here, there should be a pyrimidine here. Base pairing does not happen randomly. There is a specific rule that needs to be followed during base pairing. That's called the principle of complementarity, basically. So any new nitrogenous base cannot form hydrogen bonds with any random nitrogenous base. So a purine always forms a bond with a pyrimidine. Two purines cannot form base pairs. Two pyrimidines cannot form base pairs. You may have noticed how throughout the length of DNA, its width is uniform. It's not like DNA is wide in one region, it becomes narrow in the other, it becomes wide in the other. So its width is maintained uniformly across the length of DNA, throughout the length of DNA. What makes DNA maintain such a uniform length, uniform width throughout is the base pairing. I told you earlier that purines are slightly larger molecules. It's made up of two rings, and pyrimidines are slightly smaller, made up of just a single ring. If only you had two purines forming a base pair, that portion of the DNA would become wider. If two pyrimidines formed a base pair, that portion would become narrower. In some other place, there's a purine forming a base pair with a pyrimidine, then it would become like intermediate in width, right? But you don't see that happening in DNA. The reason why the length is maintained, the width is maintained uniformly throughout its length is because a purine, a bigger molecule, always pairs with a smaller molecule. If there's a purine on this strand, there'll be a pyrimidine on the other strand. If there's a pyrimidine here, there'll be a purine there. Okay, so it's always between purine and pyrimidine. Again, there is another rule: any purine cannot pair with any pyrimidine. A will always pair with T by means of 2 hydrogen bonds, and G will always pair with C by means of 3 hydrogen bonds. If there is A here, there will be T here. If there is A here, there will be T here. If there is G here, there will be C here. If there is G here, there will be C here. Okay, so that's how base pairing happens, and that's the reason why Chargaff's rule holds good for double-stranded DNA. If only it was a single-stranded DNA, then Chargaff's rule won't apply, because in a single-stranded DNA, since there's only one strand, there won't be base pairing. For as long as there is no base pairing, you can't say that how much of a T is there, the same amount of A will be there, right? Only if base pairing is there, that will apply true, right? Yeah, so the two strands are paired through hydrogen bonds. Between A and T, there's going to be 2 hydrogen bonds. Between G and C, there's going to be 3 hydrogen bonds. Okay? Next. The two chains are coiled in a right-handed fashion. Now, what do we mean by right-handed fashion? When we say it is coiled, we mean to say that it is spiral like that. There is a specific direction in which it is coiled. Right-handed fashion means hold your right hand up like that. This thumb will form the axis. Around the axis, in the direction the other fingers are pointing, this is the direction in which DNA is coiled. That is what it means when we say it is coiled in a right-handed fashion. Now, one complete turn of DNA, one complete turn is known as a pitch. The length of one complete turn is 3.4 nanometers. Within one complete turn or a pitch, you have roughly around 10 base pairs. And the distance between two base pairs, obviously, if in one turn you have 10 base pairs and overall the length of one full turn is 3.4 nanometers, then the distance between two consecutive base pairs will be 0.34 nanometers, right? Yeah. Hydrogen bonds, provide stability to the DNA molecule. Along with the hydrogen bonds, you can see how in DNA, the base pairs are stacked right on top of each other. Okay, so that also confers additional stability to DNA helix. Okay, so that's about the double helical DNA's salient features. Now let's talk about the packaging of DNA helix. The structure of DNA that we just discussed is the structure of naked DNA, but inside of our cells, almost always DNA exists associated with one type of protein or the other. I'll tell you the reason for why DNA does not exist as naked DNA. The reason is that our cells are super tiny, right? They measure in microns, and the length of DNA is super long. I told you that it is a very, very long molecule of a very long chain of polynucleotide is what I mentioned earlier. The length of that far exceeds the diameter of our cells. I wouldn't say it is impossible to fit DNA as it is as naked DNA inside, but if we try to do that, if it is not properly, condensed and arranged and packaged inside the cell, its functioning will be affected. Okay, so in order to make sure that the DNA, the long chain of DNA that we have, fits inside the nucleus while simultaneously retaining the ability to express itself efficiently, packaging is very, very essential. So when we speak of DNA, we talk about DNA existing in different structures, right? So inside the nucleus, sometimes you may have read that the textbook says there is DNA. Sometimes they say it's chromosomes. Sometimes you hear the word chromatin network. So what is DNA? What is chromatin network? What is chromosome? They're all essentially different forms of DNA. Chromatin is also DNA associated with proteins. Chromosome is also DNA associated with proteins. The difference is the extent of condensation. Okay, so you can think about it like this. If only you have to store threads, okay, like the sewing thread that we use for stitching, how are they generally stored at homes? Your mother, if she uses it for sewing, or anybody at home uses these threads for sewing, you know that they are wound around cardboard rolls, right? That is how they are compactly arranged. If you have like 5 different colored threads and all of them were just threads and did not have a roll, you understand how messy the whole situation would get, right? Same thing happens even in our cells. So, in order to avoid that mess, our cells very carefully and very cleverly condense the DNA so that it's more organized inside the cell. This condensation does not happen at one level. If you look at the thread and the roll of thread, there's one level of condensation where you have a thread and you have a roll, and around the roll, the thread gets wound. But in our cells, when condensation of DNA happens, essentially in eukaryotes when it happens at multiple levels. First, there's one type of protein around which DNA will bind. So that will form another thicker thread. That thread will wind again. That will form an even thicker thread. That thread will wind again, and that will form even thicker. So at multiple levels, this sort of condensation happens, and ultimately the most condensed form of DNA that we find is the chromosome. Okay. So today we're going to learn about how packaging actually happens in prokaryotes as well as eukaryotes. We'll just touch upon it. We'll not get into too many details about all of the levels. The first few levels of condensation we will talk about. If you talk about prokaryotes, you know that prokaryotes do not have a membrane-bound nucleus, right? The genetic material is not surrounded by a nuclear envelope. So the DNA in these bacterial cells and prokaryotic cells lies in a region that is known as a nucleoid. Okay, so nucleoid is the region where DNA can be found. What do we already know about DNA? We know that DNA is made up of nucleotides. Nucleotides have three components. There's a sugar, a base, and a phosphate. We know that a phosphate group is negatively charged. Because of the presence of the phosphate group in DNA nucleotides, DNA gets a net negative charge. Now, in order to wind DNA around proteins, the proteins should have the opposite charge. So, proteins that are involved in condensation will generally have a positive charge to them. In prokaryotes, you have proteins that are positively charged present in this region where DNA is found. So, if this is the region where you find DNA, the DNA that we know, the double helical form of DNA, will be associated with some proteins, okay, and it will form these sort of large loops in this region that is called the nucleoid, okay. So, it's relatively simpler compared to how packaging happens in eukaryotes. In eukaryotes, for the first few levels of condensation, you have what are called histone proteins. These histone proteins have a net positive charge. How do they get a positive charge? You have positively charged proteins, you have negatively charged proteins. How does a protein get a positive charge or a negative charge? I told you that DNA is negatively charged because it has a phosphate group that has a negative charge. What gives proteins its positive charge? What are proteins made up of? Let's get down to the basics. Proteins are made up of substances, monomer units called amino acids. So different amino acids join together to form a long chain that is called a polypeptide or a protein. Now there are 20 different types of amino acids that make up the protein. These amino acids, some of these amino acids have a positive charge, some of them have a negatively charged side chain, some of them are neutral, some of them have these aromatic rings in their side chain. So amino acids vary from each other based on what is present in its R group or its side chain. There are two amino acids, lysine and arginine. Okay, so these two amino acids in their side chain, they have positive charge. Does that mean these histone proteins are only made up of lysine and arginine? Not really. They do have other amino acids, but the amount of lysine and arginine is so high in these histone proteins that they overall get a net positive charge. Okay. Now what happens, the negatively charged DNA will coil around these positively charged histone proteins as the first level of condensation. So here, these histone proteins are of 5 different types. You have H1, H2A, H2B, H3 and H4. Among these, H2A, H2B, H3 and H4 in multiples of 2. 2 of each form what is called a histone octamer. Octa means 8. So 4 twice 2 of each will give you 8. That is called the histone octamer. That forms the core. Okay, so if this is like the histone, okay, I'll show you what a histone octamer kind of looks like. So here you can see you have H2A, H2A, H2B, H2B, H3, H3, H4 and underneath behind you have H4. So all of these will form the core. Okay, so this is called the histone octamer. Around this histone octamer, you can find DNA wound. So let's say this is one histone octamer, you have another histone octamer, and then you have another histone octamer. Around this, our DNA will wind. Okay. The DNA that winds around each histone octamer contains roughly 200 base pairs of DNA. This structure that you see here is called a nucleosome. Now, this DNA is repeatedly winding around these histone octamers. You want to ensure that they stay in place, they stay put in their place. So, there is another histone which was not part of the octamer, the H1 histone, that kind of forms, that kind of ensures, which color should we use, green maybe, that it stays put in place. This is the H1 histone. Okay. So when you look at this under an electron microscope, it pretty much looks like a string of beads. Okay. So under the electron microscope, it appears like a beads on string. The repeating units of this nucleosome is what we call the chromatin. This chromatin, when it's present inside the nucleus in the form of a network, then you call it chromatin network. This is just one level of condensation. Of course, while DNA forms the chromosomes, there are many other levels of condensation further that are made possible because of another type of proteins that are called non-histone chromosomal proteins. Non-histone chromosomal proteins or NHC proteins. Okay? Yeah. So this is our naked DNA, this is our nucleosome, this is the repeating unit of nucleosomes. This will further coil, it will further coil and becomes the chromosome. Ultimately, when the cell is undergoing cell division, during the metaphase stage, it is fully condensed. What we mean by condensation is that after every stage of condensation, the length of DNA is going to reduce and its thickness is going to increase. So if you look at this DNA here, this DNA, the width is around what? 20 nanometers, I suppose, right? Or 2 nanometers or 20 nanometers, one of those. So as it becomes condensed, this width will increase, it becomes thicker and thicker and thicker, and in metaphase it's going to be 1400 nanometers or something. It becomes thicker because of associating with proteins, and its length is going to shorten. The length will become shorter, the width will become larger. Okay, so if you think of the thread and the roll of thread, if this is the size of the thread, when it winds around a cardboard roll like that, you know how the thread, this width has increased, its length has decreased. That is what happens to our DNA also during condensation because of packaging. Okay, so that allows the DNA to fit inside the cell, to fit inside the nucleus and still express and be efficient in whatever it does. Okay, otherwise it's possible to fit. I wouldn't say it's impossible to fit a DNA inside a cell if it is not organized, if it is not condensed, but it won't be able to do its job very well. Okay, now that we know about what chromatin is, when you look under the microscope, within the nucleus, you can find some regions in the DNA, in the chromatin, where more stain is taken up. It appears darker under the microscope. And there are a few regions where the stain taken up is lighter in color. Okay, so you stain the nucleus, right? You add dyes to the nucleus, to the cell. You look at it under the microscope. There is differential staining that you can see. Some regions appear darker, some regions appear lighter. Now, the regions that appear darker are called heterochromatin. These are the two different types of chromatin. The darker regions are called heterochromatin. The lighter regions are called euchromatin. Now, why are the regions, heterochromatin regions darker? They are darker because more condensation happens there. It's more tightly coiled in those regions. And in euchromatin regions, there is coiling that has happened, condensation has happened, but coiling has not happened too much. Like the levels of coiling, if you look at, more levels of coiling has happened in heterochromatin, fewer levels of coiling has happened in euchromatin. Now, why should there be difference in how much coiling has happened inside the cell? The cell is currently existing at one particular phase in its cell cycle. Why should there be any difference in how much coiling has happened in different segments of DNA? We need to understand one thing here. Throughout the length of DNA, information is not there. Okay. So there are specific segments of DNA which carries information. There are specific regions which work as genes. And even those genes are not always required by a particular cell. Some genes are required to be expressed at a particular time, some of them are not required to be expressed at a particular time. Depending upon whether a gene is being expressed at that moment or not, its condensation pattern will change. Okay, so if a gene is not being expressed at all in that particular cell, so let's say there is no neurotransmitters that your stomach cells are producing. It's mostly produced by your brain cells, right? So if there's no neurotransmitter being produced by your stomach cell, does it have the genes of neurotransmitters? Yes, every cell in our body has the same genes, diploid cells, right? So that gene is there, but it's not expressing. So genes that are permanently switched off, that don't express, they usually are in the form of heterochromatin. It's more tightly coiled. If it has to be expressed, then it will loosen up. Okay. So even inside the cells, there are genes that are expressing in the stomach cells, but the products may not always be essential. So there is a gene whose product the cell requires, but it doesn't require it right now. It'll coil it further. Okay, so it'll condense it further. If there is a gene whose product is required at that moment by the cell, that will be loosely coiled. Okay, so depending upon the need of the cell, certain regions can be tightly coiled, certain regions can be loosely coiled. Whatever is tightly coiled at this moment, if that product, gene product is required a couple minutes later, that will uncoil and become euchromatin. Okay, so when I say the gene is expressing, it happens by means of two steps. Once the first step is called transcription and the second step is called translation. Genes can express, DNA can express via these two steps only. So the first step called transcription happens only when genes are actively being expressed. So wherever active expression is happening, there it's loosely coiled. So transcription is happening. So we call that transcriptionally active. Wherever active expression is not happening, transcription is not taking place, where it's more tightly coiled, we say that it is transcriptionally inactive. So when we're talking about the difference between heterochromatin and euchromatin, we must keep these 3 points in mind. Firstly, heterochromatin is tightly coiled, euchromatin is loosely coiled. Heterochromatin takes up most and it appears darker. Euchromatin appears lighter in comparison. Heterochromatin is not transcriptionally active, which is why it is more coiled. Euchromatin is transcriptionally active, which is why it is loosely coiled. So these three differences are important. Here you can very clearly see this is the nucleolus region, and within the nucleus you can find there are dark colored regions and lightly stained regions. Okay, so that's the difference between heterochromatin and euchromatin. Now let's start talking about the search for genetic material. There are 3 important experiments that you should remember and understand. These are pretty chill. Okay, so yeah, we'll start off with Griffith's experiment. Frederick Griffith performed this experiment called the transforming principle experiment in the year 1928. So what he did was he basically was working with 2 organisms, one a mouse, not a mouse, many mice of course, and then he worked with a species of bacterium called Streptococcus pneumoniae. Streptococcus is a bacterium, Streptococcus pneumoniae, this specific species of bacterium used to cause pneumonia in mice, and because of pneumonia the mice would die. If they ever got infected, they would die. Okay, so what he did was he grew Streptococcus pneumoniae in his lab. There are 2 strains of Streptococcus pneumoniae. One is called the rough strain, the other one is called the smooth strain. What are strains in bacteria? Let's first understand that. It's similar to how you have breeds in dogs. Dog is all one species, but you have different breeds in dogs, right? Like you have the pug, the Labrador, the golden retriever, you have a German shepherd, and you have so many other breeds. They are all the same species, but different breeds. Like that, in bacteria, you have one species and you can have very many strains, okay? So this Streptococcus pneumoniae had 2 strains. One is called the rough strain, one is called the smooth strain. Why are we calling one rough, one smooth? What significance does it hold? The reason why we're calling the rough strain is because when you grow them in a bacterial culture, when you're growing them on a plate, on a petri plate, on an agar medium, it forms colonies, right? Bacteria form colonies. You can see colonies in the form of dots on top of your culture plate. Whatever colonies formed from the R strain bacteria, they had a rough appearance, whereas the ones that were formed from smooth strain bacteria, Streptococcus pneumoniae, they had a smoother appearance. The reason why this appeared rough is because outside the cell wall, there was no capsule present, there was no other polysaccharide jelly-like layer present. As opposed to that, in the smooth strain bacteria, outside the bacterial cell, there was like a capsule present. Okay, so that's the primary difference. Because of the difference in its appearance, it's given these names rough strain and smooth strain. And yeah, apart from that, another thing is, I told you how the Streptococcus pneumoniae, as the name itself indicates, cause pneumonia in mice and end up killing them, right? So the rough strain did not have the ability to cause pneumonia, and the smooth strain had the ability to cause the disease. Because this does not have the ability to cause the disease, you call them non-virulent. This one does have the ability to cause the disease, so you call them virulent. Okay, so these are the two different strains of Streptococcus pneumoniae that Griffith was working with. Now, what exactly did he do in his experiment? This has so much drama, I tell you. He has some mice in his lab. He first takes R strain bacteria. R strain bacteria, can it cause pneumonia? It cannot. If it cannot cause a disease, obviously it will not end up killing the mouse, right? So he takes R strain bacteria and he injects that into mice. Mice is very happy. Mice are very happy. No disease, eating, sleeping, happy. What he does next, he takes S strain bacteria capable of causing disease, virulent strain, injects them into mice. Very sadist, I get it, but yeah. He did that and the mice ended up getting pneumonia and they died. Okay, then what he does, now he was sadistic with these mice and he got them killed. Next, he is sadistic with bacteria. He kills the bacteria. Okay, so he heat kills specifically S strain bacteria. S strain bacteria that were capable of causing disease, he heats them. He heats them until they die. That is called heat-killed S-strain bacteria. So he introduces or injects this dead bacteria into mice. What can dead bacteria really do? They're dead, right? They can't cause infection because all of its components would have gotten broken down, right? So it does not cause pneumonia. Dead bacteria did not cause pneumonia. They were inactive, not capable of causing infection because they themselves were dead. And the mice happily survived. Next one genius idea he does, he gets. So what he does is, look here. When he added only rough strain, mice survived. You can't see this. Okay. When he added R strain, I'm sorry, the S strain, the mice died. When he added heat killed S strain, the mice survived. Now what he does is, he will mix live R strain along with heat killed S strain. Both of them did not cause infection. He mixes the two and that mixture he will inject into bacteria. You have two types of bacteria here. Rough strain, not capable of causing disease. S strain could cause disease, but now they are heat killed, they are dead, so they can't cause disease. The mixture he injects into these mice. He hadn't expected that the mice will develop pneumonia and die. But to his surprise, the mice develop pneumonia and die. Then what happens is like even more twist and turn. They will perform like a post-mortem autopsy on this mice. And they will check the tissues of the mice, and in the mice what they find is this kind of bacteria. But had we added them at all, had we introduced them at all into the mice, we hadn't. What had we put? We'd put this, we'd put this. How did this guy come into this bacteria? How did this get into this bacteria? There's only two possibilities. Either this should have become this, or this should have become this. Rough strain, smooth strain. What is the difference? This does not have a jelly coat. This has a jelly coat. It's possible that the R strain bacteria that are alive have somehow developed the ability to form a capsule around themselves, developed the coat and become S strain. This is one possibility. What is the second possibility? These guys that already had the jacket, they were dead. They somehow miraculously came back to life. This is very, impractical to think about, because once you're dead, it's over, right? You can't come back to life, right? That only happens in Ekta Kapoor serials, but not in real life. So this you can eliminate. If it's dead, it's gone, it's over. It cannot come back to life. So what is the other possibility? More probable thing that could have happened is this one, somehow developing the ability to, produce a coat around itself, a capsule around itself and become S strain. So what he infers from this experiment is that he says these guys didn't originally have the ability to form a coat around themselves. He says that there is something in the heat-killed S strain bacteria, its genetic material. We do not know its nature. We do not know if it's carbohydrate, protein, lipid, RNA, DNA. We do not know. He says there is something in this. The something in the dead bacteria gave the live R bacteria ability to synthesize a coat. What it is, he will not say. He calls it the transforming principle, because this is transformation in the microbial world. You have seen transformation reels where people who are 110 kilos loses weight. I mean, they lose weight and they come up and they are 60 kilos, 70 kilos lighter. I mean, 70 kilos right now. So all those kind of transformation reels you would have seen, right? So this is like the transformation reel in the microbial world. So if only there was reels and Instagram in the microbes, these bacteria would have been in the top of the game. So it's like the R-strain bacteria comes over near the what do you call it? The camera, and then it's like, oh, so such a timid person. And then it does this, and then when it comes back, it's wearing a jacket and has potential. It has guns. It can kill now. So yeah, that's basically what happened here. But yeah, we did not get like conclusion of what the genetic material is. The whole reason why he performed this experiment is to find out what is the genetic material. Back then, people knew that something is getting passed on from one generation to another and all of that, but they did not know what was the chemical nature of that substance. They were trying to figure it out. A vast majority of people thought that proteins were the genetic material, because proteins are made up of 20 different amino acids. DNA is just made up of 4 different nucleotides. So if you look at the variety, there's more variety in proteins than in DNA or RNA. So around us in nature, we see so many variation. They thought that to explain this much variation, uh, maybe protein, since there is more variety, they are the genetic material. But yeah, this experiment just was like a stepping stone, but it did not conclude or give us what is the nature of the genetic material. What happened next was 3 people, uh, Avery, MacLeod, McCarty, they kind of continued Griffith's experiment. Okay, so these 3 guys. So what they did was, see, what is the problem statement now? We know something in this transformed this into this, but we don't know what that something is. It could be carbohydrate, it could be protein, it could be lipid, it could be DNA, it could be RNA, it could be anything. We want to figure that out now. So what did they do? They first took this out. From the heat-killed estrain bacteria, they separated out individual biomolecules. From this, they separated isolated carbohydrates, proteins, lipids, RNA, DNA, everything was separated from the heat-killed estrain bacteria. This is similar to what you would do if you were to, let's take this example where you have 4 red colored clothes. Okay, so you have a red dupatta, you have a red shirt, you have a red skirt, and let's say you have a red kurta, whatever. Okay, 4 different garments that are red in color. While you're washing these garments in your washing machine, let's say that you've put your white color pants along with that. Okay, so after the washing machine has run, you look for your white colored shirt or pant or whatever you put. The white color fragment is no longer white color, that is also turned red. But you do not know which of those red colored garments release the color so that the white is turned into red. So what will you do if you want to find out which of the 4 red garments was responsible for the white shirt turning into red? You will try to soak the red color garment separately along with a small white piece handkerchief, something that you have at home, to find out which is the culprit, right? This is exactly what these guys did too. Very, very smart of them to think that way. So they kind of took this and separated out all of its individual components. So they had sugars separated out, RNA, proteins, and DNA separated out. Lipids are not a part of this here because nobody really believed that lipids could be the genetic material. Mostly the forerunners in the race for who is the genetic material were sugars, proteins, and mostly proteins, DNA and RNA. So they separated that out. In this experiment, no mice are harmed. Okay, only bacteria they're working with. So they separated out these components, and to these separated out components of the smooth strain heat killed bacteria, they will add R strain bacteria that are non-virulent, no capsule. Okay, so they will allow it to grow for some time in whichever test tube or wherever they were growing it, in whichever culture. Sugar was there, RNA was there, protein was there. They will not find any estrain bacteria formed. Transformation has not happened. But only in the test tube where DNA of the heat-killed estrain bacteria were mixed along with the live R strain, they will find live estrain bacteria. This kind of confirms that DNA itself is the genetic material, but people back then were like so adamant and they did not want to buy this. And they're like, oh, you've not separated the DNA properly. DNA does not exist as DNA in most cases, like I mentioned earlier too. It's associated with proteins. You've not separated it out properly. Probably some proteins have gotten along with your DNA sample, and it is not the DNA but the proteins along with it that is causing this, and so much drama happened. Not many people approved of these results. They did not accept it. And further, two more people, Alfred Hershey and Martha Chase, they both performed another experiment. Okay, so that experiment kind of gave us like the final proof that DNA itself is the genetic material. Okay, yeah, so before that I forgot to mention this. Another way in which this experiment was confirmed, the results of this experiment was confirmed, was they take heat-killed strain bacteria, they remove the lipid and sugar. Major contenders are proteins, RNA and DNA. So they separate that out into 3 different test tubes. So what is present in these test tubes? You have heat-killed strain bacteria, but you do not have its sugar and lipids. You have RNA, DNA, protein mixture. To one test tube, they add protease, which is a protein digesting enzyme. To the other, they add RNase, which is an RNA digesting enzyme. And to another one, they will add DNase, which is a DNA digesting enzyme. So there what they did was, they added live R strain bacteria to these cultures. R strain bacteria are added there. And they checked for in which test tube transformation will happen. In the test tube that had DNase, DNA will be broken down. Whenever DNA has been broken down, transformation did not happen. When DNA was intact, in this case proteins were broken down, in this case RNA was broken down. Despite RNA being broken down, protein being disintegrated, despite that, transformation happened, because in both these cases DNA was untouched, unharmed, right? So transformation happened. But over here DNA itself was broken down, so transformation did not happen. This again is proof enough that DNA is a genetic material, but people, right? Yeah. So yeah, this is the Hershey and Chase experiment. This was performed by 2 people. This is a cool experiment too. So what they basically did here was, they played with 2 organisms. One is a bacterium, one is a virus. This virus is called a T2 bacteriophage. It infects a bacterium called E. coli. Okay, so the 2 organisms that are used here are a bacterium called E. coli that is found in the colon, and then you have, and a virus called T2 bacteriophage. Okay, so this T2 bacteriophage, they cultured in the lab. Okay, so they grew it on 2 separate media. Okay, now whenever you're growing microbes in the lab, you provide them with food to eat in the form of a nutrient medium. Whatever you provide, that only they should eat. Now in the medium where I'm growing these viruses, I'm providing them with a source of carbon, a source of phosphorus, a source of sulfur, a source of nitrogen, and all of these things. Okay, so I create 2 separate types of media here to grow 2 different sets of T2 bacteriophage only. They themselves do not have any differences. It's not like one is like R strain, one is like S strain. They are both the same, but the culture medium that I am using to grow them is different. I'm giving them 2 different kinds of food. You can think about it like that. Now, in one culture medium, I've added radioactive phosphorus. In the other one, I've added radioactive sulfur. We've already discussed biological classification. You already know the structure of viruses. Viruses are very simple in structure. They have just 2 components. They have what is called the genetic material. In this case, bacteriophage has the genetic material DNA. And surrounding the genetic material, they have a protective protein coat. Okay, so there is a protein coat and there is DNA. In one medium, they have added radioactive sulfur. In the other one, they've added radioactive phosphorus. Okay. What we must remember here is that in DNA, phosphate group is there, so phosphorus is there. In DNA, sulfur does not exist. In proteins, which are made up of amino acids, phosphorus does not exist. But certain amino acids like cysteine and methionine do contain sulfur. Okay. So this radioactive sulfur will get incorporated into the protein coat in this case, because DNA does not have sulfur, DNA will not become radioactive. Here, the DNA, this virus is being grown in a medium containing radioactive phosphorus, that is like food for it. So whatever is available, that is what gets into its body. Whatever is into its body, that is what it will use up to synthesize newer molecules. So if it were to make a new molecule of DNA, it requires phosphorus. Where does it take the phosphorus from? It takes it from the medium. What phosphorus is available there? It's P32 radioactive isotope of phosphorus. That's what is available there. So it will take it up and incorporate that into its DNA. So in this case, the DNA is radioactive. Let me show that in red color. Okay, so the DNA here is radioactive, but protein coat is normal. In this case, the protein coat is radioactive, but the DNA inside is normal because sulfur is radio-labeled. Here DNA doesn't even have sulfur, so it will not incorporate it. This is the first uh, part of the experiment. Okay, now that you have two subsets of T2 bacteriophage available for your experiment, we move on to the actual experiment where we have two sets of bacteria, E. coli growing. These are very normal. There is no change in the medium that it is growing. All of these bacteria are grown in a normal medium containing normal isotopes of sulfur and phosphorus. You just have two such sets. They are grown on normal media. There is no difference between this bacterium and this bacterium. Both the bacteria are the same. Okay. Now, we will introduce or infect one set of bacteria in one culture medium with with the virus that has radioactive protein coat. The other one we will infect with virus that has radioactive DNA. Okay, so the first step here is infection. We cannot see this. Remember, this diagram we can draw after knowing the results, but when they were performing the experiment, they could not look at the bacterial cells under the microscope and say like, oh, protein coat is sitting outside, DNA is going inside. If only we knew all of that, we could have directly told that DNA itself is a genetic material. The problem is that the molecules are too tiny, so you can't look at them under the microscope. Viruses are too small, you can't see them going and sitting on top of the bacteria, injecting its DNA. All of that we know today from these experiments, but back then they did not see this happen real time. Okay, so they just performed their experiments. They just infected this, and in the next step, they wanted to make sure that if there is any component of the virus that has not entered the bacterial cell, let's just separate them out. So what they did in the next step was called blending. Blending is like running your mixer to make your fruit juices, very similar to that. There is a blender where they will kind of spin this whole thing. If at all there is any part of the virus that has not entered the bacteria, if there is any virus that has not entered the bacteria, virus or its particle that has not entered the bacteria, they just wanted to separate it out of the bacteria. In the third step, we perform centrifugation. In centrifugation, there is an instrument called a centrifuge where you will insert these centrifuge tubes, and this centrifuge kind of spins at very high speeds, as much as 75 to 1 lakh rotations per minute. So they took these mixtures of bacteria along with bacteriophages, two separate centrifuge tubes. They spun it in a centrifuge at very high speeds for a long period of time. In a centrifuge, what happens? Because of centrifugal force, the denser particles will settle down and the lighter particles will stay on top, right? Think about it like you have a glass of water and then you mix it with sand. Sand will have different size of sand grains. The bigger sand grains will settle down at the bottom because of its density, and the smaller ones will kind of be retained on top, and they will also settle down eventually, but it will take a longer time for the lighter particles to settle down. Same thing even here. What are the components that we have in the centrifuge tube right now? What we have, what we have here is within this mixture is bacteria along with viruses. We don't know anything yet. We have not seen anything yet. Okay. You have bacteria, you have virus. If you compare the density of bacteria and viruses, obviously bacteria are much larger, right? So ideally, bacteria have to form the pellet, right? Bacteria forms the pellet, and in the supernatant on top, the supernatant that you find on top, you will have the virus present. So that is the idea, because that is lighter, that should ideally be present in the supernatant. Now, when they check these centrifuge tubes for presence of radioactivity, what they will notice is that in whichever tube we had introduced bacteria along with viruses that had the protein coat radio labeled, they will see radioactivity in the supernatant. What do we already know? In this case, the protein was radio labeled. In this case, they will observe radioactivity in the pellet. We already know that in the pellet, bacteria is there. What spelling of pellet have I written? Okay. So over here, the bacteria are present. Were the bacteria themselves radioactive by any chance? Not really. They were grown on very normal media. So they themselves did not have radioactivity. We know that they themselves were not radioactive. We know that they were infected with viruses that had DNA that was radioactive. So if the bacteria here are showing radioactivity, what does that mean? The component that was radioactive in the virus has entered into the bacteria. That's what it conveys, right? So whichever is the genetic material, that is what will enter the host, right? That is how the expression or passing down of information, genetic information happens. So if the protein coat has remained outside and DNA has entered inside, that means DNA itself is the genetic material. So this experiment gave us the unequivocal proof that DNA itself is the genetic material. So this kind of gave us the final proof, and everybody calmed down after that. And they were like, okay, fine, we get it. DNA itself is the genetic material. Let's bow down to DNA. All right. So that's what happened. And yeah, again, let's talk about what are some properties that a genetic material, that a molecule should have in order to be a genetic material. Of course, there were many people who thought that proteins were the genetic material, or carbohydrates are the genetic material. Some of them even thought RNA and DNA is the genetic material. In order for a molecule to be considered as a genetic material, it should have certain properties, certain features. Okay, like for example, if I have to call you a very good student, then you should display some features. You should watch every lecture of mine. You should solve all the DPP questions given in class, and whenever you have doubts, you must always ask them and get it clarified. And you should be very focused and disciplined, and whatever else. Okay, so these are some features that an ideal student, a good student, should have. Like that, what are some features that a genetic material should have? Firstly, most importantly, it should be able to make a copy of itself. Why should it make a copy of itself? If I were to pass on my genetic material to my son, if I pass away whatever information I have, what will I be left with? I will be left with no information. For me to survive, for me to exist, for me to function the way I do, I need genetic material, right? I can't give away what I have. If I do that, then I will cease to exist, right? That should not happen. If an organism is reproducing, passing down its genetic material to the next generation, I should make a copy of what I have and then pass it on. Otherwise, the parent will cease to exist, right? They will not be able to function normally. So, for a genetic material, for a molecule to be considered a genetic material, first of all, it should know how to make a copy of itself. Okay, so it should be able to replicate itself. Proteins fail here. Proteins can't make copies of themselves. If a protein has to form, the information has to come from DNA only. Carbohydrates cannot replicate themselves. It's like they cannot make a copy of themselves. But DNA and RNA both are too good at that. Okay. So DNA and RNA pass this test. So the further points will apply only to DNA and RNA. In the first point itself, protein and carbohydrates are out of the race. Next, it should be stable chemically and structurally. What is the function of genetic material? It pretty much controls everything that's happening inside of your cell. So it has to be stable both chemically and structurally. You think about it like this: if it's like a blueprint or the rules of a game, let's say. Okay, so today you say that you're playing cricket and you say if the ball hits the building, then you're out. Okay, so tomorrow you change the rules again. The games, the whoever the players are, they will be like super confused, right? Every day the game rules are changing, or you have a blueprint for a certain procedure. Every day if the blueprint is changing, people will not be able to uh, you know adapt according to that very quickly. So a constant or a stable set of rules and conditions are required, right? Same thing goes here. Game is like a very um, oversimplified example that I told you. Genes are even more important. Your genetic material is even more important. How tall you grow, what texture is your hair, what color is your skin, what proteins are produced when, how are the enzymes getting across the plasma membrane. All of these, like every little thing that happens inside of your cell, it's controlled by your genetic material. It has control. It is like the control center for all the things that are happening inside your body. If such a thing keeps on changing every little minute or second, like it is PMSing, then the whole uh, you know uh, functioning of the body itself will be affected. This has very precious information that shouldn't be changing so often, or the cells will fail to function normally, right? So it's very essential that a molecule that we consider as a genetic material should not only be chemically stable, it also should show structural stability. Structural stability means it should not break off very often. So it should be structurally also very stable. So that's the second thing. Thirdly, Just because it should be stable, just because it has to be constant for the most part, doesn't mean it should not be able to change at all also. Like you have some fixed set of rules in cricket, let's say. Earlier there was no T20, but people's attention span started to reduce. According to the changes, the rules also have to bend a little, be tweaked a little. According to time, according to how time passes, it should not be too rigid also, right? Same goes for our genetic material. If the genetic material is rigid the same way, newer species will not form. We would not have evolved from apes at all. How did apes become humans? Of course, there are many ancestors in between. You had Dryopithecus, Australopithecus, and Homo habilis and Homo erectus, and all of these guys before us. How did one human ancestor give rise to another human ancestor? That's because of changes in our DNA. If changes in DNA does not happen, evolution will not happen, right? So, of course, once in a while, little scope should be there for changes in DNA also that can happen through mutation. So, such scope there should be, there should be place for such changes, but it should happen like slowly over a period of time. It's not like very frequently, it shouldn't happen. Fourth, it should be able to express itself in the form of Mendelian characters. In this batch, we've already discussed principles of inheritance and variation, so you know what we are talking about. So similar to how Mendelian characters, according to the rules of Mendel, get transmitted from one generation to another, the genetic material also should get transmitted like that. Okay, so it should be able to express itself in the form of Mendelian characters. So these are the four properties. Now we know proteins and carbohydrates have they've left the list, they've left the race and failed in the first point itself. We will compare RNA and DNA, which fulfilled the other three categories, other three criteria. So first, ability to generate replica, both RNA, DNA do it phenomenally well. Second, chemical and structural stability. Of course, they're both stable, but in comparison, RNA is a little less stable compared to DNA. There are three reasons for this. Firstly, why RNA is a little unstable compared to DNA is because, yeah, RNA has it performs catalytic activity. It is involved in catalyzing peptide bond formation. During splicing, it plays an important role. So there are some places where RNA acts like a catalyst, acts like an enzyme, which makes it a little bit more reactive compared to DNA. DNA does no such extra fancy work. Okay, so comparatively, DNA is a little more stable. Secondly, in RNA, you have free 2' OH group, which makes it more reactive. Thirdly, you have uracil in the place of thymine. Thymine has more stability compared to uracil. And also, in most cases, RNA is single-stranded, DNA is double-stranded. Being single-stranded also makes it a little less stable compared to DNA. Okay. So, stability-wise, in comparison, DNA is more stable than RNA. Whether it provides the scope for slow changes required for evolution, whether mutation can happen. Yes, in both cases, mutation can happen. It does happen all the time. But in RNA, it's much higher compared to DNA because obviously it is a little less stable, which is why more mutation happens there. One example that I always give when I talk about this is how when there was this COVID outbreak and people were talking about the different variants of the coronavirus, there was a Delta variant, Omicron variant. Within a few months span itself, the same virus used to mutate and give rise to newer variants. How was that possible? Coronavirus is an RNA virus. So RNA viruses mutate at a very fast rate. You don't see that happening so often with organisms that contain DNA as a genetic material. Even for the viruses that cause common cold, they also mutate at a very fast rate because they are RNA viruses. Okay, so that's about RNA and DNA. Both can undergo mutation. But in RNA, mutation rate is more. Mutation rate is more means stability is less. That's why. Okay. Next, it should be able to express itself in the form of Mendelian characters. Both can do that. DNA can do it only via RNA, but RNA can directly do it. So, considering all of this, if we were to ask why is that DNA is the most dominant genetic material in the living world? Except for a few viruses, majority of organisms have DNA as the genetic material. Why is that so? That's because DNA is more stable compared to RNA, and that is why DNA is the chosen one to be the genetic material over the course of evolution. But if you have to talk about which was the first genetic material to ever exist on our planet, when life evolved on Earth, on Earth, on Earth, on Earth. The first genetic material that had formed was RNA. You know, in evolution, you will learn that hot soup of the ocean, and then life evolved, coacervates, and all of that, right? So when life first evolved, the first living form came on this planet, not formed on this planet. The genetic material that it had was RNA. But because RNA is so unstable, RNA itself got evolved into DNA, and DNA, because of its better stability, has continued to exist as the genetic material. Okay? Yeah, so that's about RNA versus DNA. Now we'll get to the most important part of the chapter, which a lot of students are, afraid of. So we will talk about the central dogma of molecular biology. We will talk about how exactly DNA controls everything, and we will just fall in love with DNA. Okay, so let's start. Well, before we move on, let me sprinkle some magical dust all over you so that you gain the ability to comprehend whatever we're going to learn moving forward. Because what we're going to learn now is one of the most dreaded concepts in this chapter. Many students fear this. So, hopefully, if you all promise to give me your undivided attention and focus, hopefully, by the end of this chapter, you will feel better. You will feel better about this whole concept. Okay, so let's go. What is central dogma of molecular biology? This was basically proposed by the same Francis Crick, who was involved in elucidating the structure of DNA. So he says that information in DNA is expressed via two steps unidirectionally in the same direction. That's what he proposed. So let's try and understand this. Okay. Let's consider one of our cells. For example, this is a eukaryotic cell. Eukaryotic cells have a well-defined nuclear membrane. Right within the nuclear membrane is our hero DNA molecule sitting and chilling. And then, yeah, in different segments of DNA, you have information for different things. So what do we know? We know our DNA is a double-stranded molecule. And this DNA is made up of what? Nucleotide. Right? So, we say that DNA is the head of the cell, it is the control center, it pretty much controls every little aspect of the cell. All of the functions are coordinated by DNA, and all of that we talk about a lot, right? But if you think about it, unless the cell is dividing, unless the cell is in M phase, the DNA doesn't really leave the nucleus much, right? It doesn't leave the nucleus at all. Only when the cell is dividing, the nuclear membrane disappears, DNA gets condensed into chromosomes, aligns at the center of the cell, metaphase plate, and all that drama happens. But otherwise, the cell, when it's in interphase, the DNA is pretty much to itself sitting inside the nucleus. Despite being so distant, like physically separated from the cytoplasm, how is DNA capable of, managing all of the activities that are taking place outside the nucleus? We see it's the control center, it takes care of everything that's happening inside the cell, but it is not leaving the nucleus at all. How does it do so? You can think about it as the dean of your school or the principal of your school or the headmaster, whoever is like the top guy in your school. You can think about it like that. Okay. They may be managing all of the activities that are happening in the school, but they themselves are not doing everything. They're not cleaning the school, they're not teaching you physics, math, biology, and everything, and they're not helping you perform experiments. So, different things, they have different people employed, right? A physics teacher is an expert in physics, they will teach you physics. A biology teacher teaches you biology, the lab assistant helps you perform experiments, the housekeeping stuff. Make sure that the whole institution, the campus is maintained neat and tidy. So, they have different people to perform different things. Likewise, this being molecule sitting inside the nucleus, this by itself does not do anything. It has many proteins that will do the job that is required to be done. But where do proteins form? Proteins form in ribosomes. Ribosomes are called protein factories. If this is like an endoplasmic reticulum, ribosomes may be present on top of it, studded on its surface, or they may even be present in the cytoplasm. Regardless, ribosomes is where proteins get synthesized. Now, the information to synthesize proteins is present in DNA. Different segments have different information for different proteins. Let's assume the cell right now requires a protein, and the information for the synthesis of that protein is present, let's say here. Okay, so this DNA has information for the synthesis of a particular protein. DNA doesn't get out of the nucleus, ribosome does not get inside the nucleus. So who is going to carry this information from DNA, copy it and go and pass it on to the ribosome? You need a molecule that can copy that information and go pass it on to the ribosome, because they both are physically, spatially separated, right? The nucleus, the DNA is in a different place, the ribosome is in a different place. So over here, the molecule that helps copy information from DNA and take it to ribosome is RNA. It's a molecule of RNA. Okay, so the message in DNA is copied on to a strand of RNA, which is also made up of nucleotides. This process by which information gets transferred or copied is called transcription. Remember, I told you transcriptionally active, transcriptionally inactive. This is exactly what I was talking about. So now the information got copied. Now this RNA goes to the ribosome. This ribosome reads the message present in RNA and it will produce a protein. What is a protein? Protein is a long chain of amino acids, right? They are polypeptides. So reading the message on RNA, a polypeptide chain will form. This process by which protein is synthesized is called protein synthesis. Polypeptides are made up of amino acids. And protein synthesis is also called translation. Why should we call it translation? What is the translation that we know? Whenever two people who are speaking two different foreign languages, they don't understand each other's language, then you need translator, right? If you want to understand the meaning of some words in some other language that you don't understand, you will need a translator. Like I do not understand Malayalam. If I have to make sense of something that is given to me in Malayalam, or I listen to something in Malayalam and I want to find out what it means, I will just type that and ask Google to translate it. Right. So we use translator when we do not know some language and we want to understand the meaning of it. So even over here, translation is happening because communication is happening between two different languages. Okay. So one thing here is DNA getting copied to RNA transcription. DNA speaks the language of nucleotides, RNA speaks the language of nucleotides. Both of them understand each other just like you and I are understanding each other right now. They speak the same language, no problem. Next, RNA has to form polypeptide. Information in RNA has to be, used to synthesize proteins. Proteins speak the language of amino acid. RNA speaks the language of nucleotides. It's like a Chinese person speaking with a Kannadiga like me. I can understand Kannada, I can speak in Kannada. I do not know the head and tail of Chinese, and the Chinese person that I'm trying to communicate with does not know Kannada. Absolutely. So what do we need in this case? We need a translator that understands both languages, can speak both languages. Whatever the Chinese person says, he will have to understand that, translate that to Kannada and convey it to me. Whatever the Kannada person, whatever I say, this person will have to translate that and tell it to the Chinese person, right? So a translator should understand both languages. When translation happens, there is one such molecule which is bilingual. It can understand the language of nucleotides, it can understand the language of amino acids, and kind of acts like a translator and makes the whole process happen. That's why we call this translation. So in this chapter, we're going to learn about replication, transcription, translation, all of that in detail. So brace yourselves. We're going to start with replication. But before that, I want to point out one thing. DNA making a copy of itself is called replication. We learn about this process. Also, we will learn about how DNA gets copied onto RNA by a process called transcription. And then we will learn about translation too. Francis Crick mentioned that expression of DNA or transfer of genetic information happens from DNA to RNA to protein in the exact same order. That holds good for most cases, but in the case of viruses that have RNA as a genetic material, virus infects hosts that are eukaryotic. In all eukaryotes, eukaryotes and prokaryotes, organisms that are not viruses, like in bacteria and eukaryotes, DNA itself is the genetic material. If it has genetic material, RNA, and it's trying to infect a host that has DNA as the genetic material, the host will not be able to make sense of that RNA genetic material. In order for the virus to make use of the host cell's machinery, it should first be able to convert its RNA into DNA so that the host cell's machinery understands what to do with the DNA, right? So, in case an RNA virus is infecting a host, the first thing that it will have to do is copy RNA information, the information present in RNA to DNA. This happens where? Only in viruses containing RNA as a genetic material, not in all viruses, only in RNA viruses. And this process is called reverse transcription. This is called reverse transcription. Yeah, because the exact opposite of transcription is happening here, so it's called reverse transcription. Let's start by talking about replication. Replication, as the name itself indicates, making a replica or the exact copy. We are speaking of DNA replication here, how DNA makes an extra copy of itself. Before it divides, before it undergoes cell division, all of these cells will make an extra copy and then only divide, right? We've learned about that in cell cycle and cell division. So what happens here? Many people believed that DNA replication happens in different modes. Some people thought it was conservative, like DNA by itself has two strands. The two strands of DNA, according to what some people believed, was that both these strands will go to one daughter cell and the other two newly formed strands will form in the other DNA molecule. When we're talking about DNA replication, you have two strands. This has to get replicated and two new strands have to form, right? Some people thought that both the existing strands will be retained as within the same DNA molecule and two brand new strands will form. That was called the conservative mode. Secondly, some people thought that it happens semi-conservatively. What that means is, you have two strands in the existing DNA molecule. One of these strands ends up in one DNA molecule, the other one will end up in the other DNA molecule. In both DNA molecules, you have a brand new strand. Okay. So this was called semi-conservative because conservative means you're conserving something. Here, completely both the strands were conserved in one molecule and the other one was brand new. Here, both of them are partially conserving it, semi. So this was called semi-conservative, and there was another mode of replication that people thought that's how replication happened. That was called dispersive, where randomly different segments of DNA will have new, a little bit of old DNA, that way. Okay. Ultimately, we got to know that replication of DNA happens semi-conservatively. How did we arrive at the conclusion that replication happens semi-conservatively? That was because of an experiment that was performed by two people, Messelson and Stahl. Okay, so they performed this experiment in the year 1958. They were working on the bacteria E. coli. Like I mentioned earlier, when you're growing bacteria in the lab, you provide it with the best of conditions. Make sure that it is experiencing a resort-like condition, so that when it's happy, when everything is provided perfectly, when the conditions are optimum, it's going to grow better. Okay, so it's good for you, right? So we provide it with food, we provide it with the right kind of temperature, all the conditions that are required for the best growth of bacteria. So we provide it with food. So what they did when they were performing this experiment was, they took in their nutrient medium, let's say if this is the nutrient medium in which they are growing their bacteria, within this nutrient medium, they added a nitrogen, NH4Cl, that had the heavier isotope of nitrogen. Normal nitrogen is N14. The heavier isotope is N15. Now, if I'm providing nitrogen source in the form of ammonium chloride to the bacteria, that is what they will make use of during their protein synthesis. That is what they will make use of during their DNA synthesis. In DNA, you have nitrogenous bases which require nitrogen. In proteins, you have an amine group which requires nitrogen. So, for all of its nitrogen needs, it will make use of the same N15 because that is what is available to it. That's the food that we're feeding it. Whatever is available, that is what they will take up into their cells, and that is what they will incorporate into their biomolecules. We've spoken of isotopes in the past also when we discussed Hershey and Chase's experiment, but over there it was not a heavier isotope, that was a radioactive isotope. This is different. Here you cannot detect radioactivity. Here you can make out the difference between N14 and N15 only by performing ultracentrifugation, not otherwise. Okay, we'll get to that in a little while. For now, just understand they grew bacteria, E. coli bacteria in a medium. Of course, there was a carbon source, sulfur source, all of that. But along with that, there was a nitrogen source, and this nitrogen was a heavier isotope of nitrogen, N15. Remember that. So all these bacteria got was N15 for several generations, and every time they underwent cell division, they have to make an extra copy of DNA. So for the new DNA, they need nitrogenous bases. For the nitrogenous bases, they need nitrogen. And what nitrogen do they have? They have N15 nitrogen. That is what they incorporated into their DNAs generation after generation. They provided this N15 nutrient medium up until a point they were kind of confirmed that all the bacteria that are growing here have N15 DNA only incorporated, N15 nitrogen only incorporated in their DNA. Once they were sure of that, they transferred these bacteria into another medium. Let's say these are the bacteria that were growing there. They transferred these bacteria into another medium that now contained N14. See, E. coli, okay, it undergoes cell division once in every 20 minutes. That's the frequency. So after they were sure of all bacteria having N15 DNA, they took it and transferred it to a medium containing N14 nitrogen. Did I say N15 DNA? Yeah, this is N14 nitrogen. Now what happens? Now after 20 minutes, it has to undergo cell division. If it has to undergo cell division before that, within the cell cycle, they have to make an extra copy of DNA. For that, they require nitrogenous bases. For that, they require nitrogen. What nitrogen is available to them now? All they have here is N14. So whatever new has to form, that will utilize N14. Whatever existing DNA is there, that will remain N15. Newer DNA, newer strands that need to form, for that N14 is available. So new N14 is not available here. So whatever new strands form, all of them will be N14 DNA. So after transferring bacteria from here to here, for every 20-minute intervals, they will, take out DNA, take the microbe, bacteria, isolate, put them into centrifugation tubes and spin it in a ultracentrifuge. Okay. So here they use a type of centrifugation method that is called cesium chloride density gradient ultracentrifugation, where you already have a gradient established inside the centrifugation tube. It is a mixture or it's a solution of cesium chloride already here. This cesium chloride, according to its density, would have already formed a gradient where there is more dense particles here, less dense particles here, even less dense particles here. Okay. Into the cesium chloride solution, we are adding our cells and spinning it. Okay. Now, according to the density of DNA, bands will form after centrifugation. The number of bands indicate something important. If there's only one band formed, that means all of the bacteria in that sample have the same kind of DNA, same density of DNA. If two bands form, that means within the sample you have two types of DNA varying in density. Okay, that is one thing. How many bands are formed is important. Secondly, the location of band formation is also important. If bands are forming lower, that means they are more dense. If they are forming slightly higher, that means they are relatively less dense. Okay. So when they spin it, before transferring, they will obtain only one band which is slightly lower towards the base, indicating that it is of high density. They are sure that this is N15. This is the standard, they know about this. After transferring it to a medium, 20 minutes later, if they spin it in a centrifuge, again perform the same cesium chloride density gradient ultracentrifugation, they will notice that a single band will form. This single band will form slightly higher in position compared to this. So this indicates that this bacteria has only one type of DNA, these bacteria have only one type of DNA, but that DNA is lower in density compared to this. Had it been conservative mode of replication, this should have formed two types of DNA. This will remain as it is and another one will form, right? This would have happened had it been conservative. If two different types of DNA are there, at this point we should have gotten two different bands. But did we get two different bands? We didn't. That kind of disproves that conservative mode of replication does not happen. Here itself we kind of got proof that the mode of replication is semi-conservative. Only if it is semi-conservative you will get this single band and it will be slightly higher than this. Furthermore, to confirm their results, they will allow the bacteria to, after 20 more minutes, they will take out the bacterial sample and then spin it again. 40 minutes from here. After here, 20 minutes. Again from here to here, 20 minutes. Because that is the duration the bacteria take to divide. Okay, so that's how frequently it divides. I think I mentioned that earlier also. Yeah. So after 20 more minutes, what happens is, here they will see two band formation. Two band formation means there are two different types of DNA. With respect to their density, there are two different types. The number of red color strands will not increase any further because there is no more N15 nitrogen available to them. Whatever is available to them is N14. So whatever new strands have to form, that will be N14 only. Even after many generations, the number of red color strands, the number of hybrid DNA will not increase. That will stay the same. Newer and newer DNA molecules that will form will all contain lighter DNA, N14, N14, because that is what is available to them. Here, the formation of two bands happens. One band will be exactly in the same position as this band, indicating that you still have hybrid DNA here. Another band will be formed slightly higher than that, which indicates that you have slightly even less denser DNA here. So that kind of confirms that when it was the heaviest bottommost band is formed, both are heavy strands. So that is heavy DNA. One generation later, after transferring into N14 containing medium, you have one band. At that point, all the red strands would have separated and new blue strands would have formed. So in each DNA molecule, you have one DNA strand containing N15, another strand containing N14. We call that hybrid DNA. Since this has one strand N14, which is slightly denser, less dense than N15, this density will be lesser than this. So it will be slightly higher. And same thing logic you apply here, this will remain as it is. So this band will remain. Newer DNA molecules have both strands N14. They are light DNA molecules, so they will form a band slightly higher. So this kind of proves that replication of DNA happens semi-conservatively. Okay. Now, similar experiments were performed by Taylor and his colleagues in the same year 1958. They make use of a eukaryotic organism, a plant called faba beans. Its scientific name is Vicia faba. They make use of radioactive thymidine here and they also end up getting the same results. So it is proven that replication happens semi-conservatively. So how exactly does this replication happen? What is the process? All of that we have to learn now. Okay. So we will learn about how replication takes place in prokaryotes, because eukaryotic replication is a little more complex. In prokaryotes, it's relatively simpler. So we learn about how it happens in prokaryotes. Okay. So let's take a bacterial cell, for example. If this is like a bacterial cell, you know that there is a region inside the bacteria called the nucleoid where the DNA is present like this. DNA in bacteria is circular. So if you kind of zoom into this, the DNA is going to look somewhat like this, I would say, right? Okay, poorly drawn, but look, you get the point, right? So you have this DNA molecule. When replication has to happen, the two strands of DNA have to separate. It cannot happen anywhere. There is a specific region in DNA where the separation happens, and that region is called the origin of replication. So let's say this region of DNA, there's a particular nucleotide sequence which indicates when replication happens, that is where it needs to start. That sequence is called origin of replication. It's also called ORI site. So we will focus on this origin of replication and I'll show you what happens there. So over there, the hydrogen bonds between the two strands of DNA, the hydrogen bonds between the nitrogenous bases, that will separate. Okay. So this hydrogen bond breaking is brought about by an enzyme called unwindase or helicase. From this point, replication will begin and it will proceed in both directions like this. That is the direction of replication. Ultimately, it starts from here, it goes till here, and by the time they reach this point, two molecules of DNA would have formed. Okay. So now, this bubble sort of thing that has formed has a name. We call it the replication bubble. This replication bubble has two replication forks. This is one fork, this is one fork. I will tell you what happens on one side of the replication fork. Okay. On the other side, the exact same thing will happen. Whatever happens here, same thing will happen here. Okay. So these are called replication forks. In your textbook, directly replication fork is mentioned. They've not mentioned replication bubble. Just so you understand where this fork-like structure is coming from, I've shown you this replication bubble. Okay. So let's see what happens during replication. So firstly, the two strands have to separate. The principle that governs the replication process is the principle of complementarity. We know what complementarity is. We know what complementary base pairs are. Okay. So let's say I've taken orange color DNA. Let's maintain that. So let's say this portion of DNA, reflection, not able to see, right? Hope it's TACT. Let's say this is the sequence that was there in DNA. After helicase has acted upon this DNA, the two strands have separated. Okay. Have A, T, G, C, T, A, C here. Now, a new strand will form complementary to the existing strand from the 5' direction to the 3' direction in both cases. There is an enzyme called DNA dependent DNA polymerase. So what this basically does is, it reads that there is an A here, complementary to that it will add a T. It reads there is a T here, it will add a, sorry, what was I thinking? Okay, likewise, it will add C, T, T, A here. Now look at this molecule, this molecule, this molecule, they all are alike. We had one molecule, now we have two molecules. Okay, so we got two identical copies of DNA. Each molecule has one parental strand and each molecule has one new strand. That is how semi-conservative mode of replication works. In this case, the existing parent strands are called template strands. I told you that DNA dependent DNA polymerase will read whatever is present in the template strand and add a complementary nucleotide, right? And forms the long chain, elongates the chain. Why are we calling this DNA dependent DNA polymerase? It's obvious why you call it the DNA polymerase, because it is polymerizing the DNA chain. It's adding more and more nucleotides and making it a long polymer. That is obvious. Why should we call it DNA dependent DNA polymerase? Because this DNA polymerase cannot function if this template does not exist. It can work only based on a given template sequence. If it reads there is a T, it can add A, but without this T, it does not know what to do. So its functioning depends on a template, which is DNA. So you call it DNA dependent DNA polymerase. Okay. So this is the principle that governs replication of DNA. Now, when you look at this replication fork, you can see that on one strand, on this strand, blue color lines represent the newly synthesized strands, the orange one represent the existing strands. You can see that in one strand, synthesis of DNA is continuous, but on the other strand it is kind of staggered, like it is happening in small arrows are represented. On one there is continuous arrow. Why is it this way? Why can't you represent this also continuously, or why can't you represent this also like this? Why is there a difference here? We'll try and understand that. When we talk about this DNA dependent DNA polymerase, I mentioned how this is what is responsible for the synthesis of the new strand. This is what reads the nucleotides present on the template, and corresponding nucleotides are added in the new chain. But this DNA dependent DNA polymerase can function only in 5' to 3' direction. When replication happens, there are a lot of enzymes involved, all of which are not mentioned in your syllabus. When replication happens, enzymes, which color? Orange is. Enzymes like DNA dependent DNA polymerase, they are sitting here. Okay. Sitting there only, not like literally sitting, but you get my point, right? They are kind of localized there. They can add new nucleotides only to the 3' OH end. I told you when we were discussing the phosphodiester bond formation that newer nucleotides can only get added at the 3' end, right? That is how the chain seems to elongate from 5' to 3' end, because at the 3' end newer nucleotides are getting added. So this has the ability to add nucleotides only at the 3' end. Now, if you look at this image, if this enzyme is sitting here, 3' end of the new strand is closest here to it. So it can easily keep on adding newer nucleotides here. So continuously newer nucleotides get added because it's closer here. But on the other strand, because of anti-parallel nature of DNA, where is the 3' end? It's here. This cannot jump here and add nucleotides at the 3' end, right? So it's impossible. This is localized here, right? So what it does is, it'll pull a small portion of this. So it'll pull like a small portion of this template strand towards it. There will be other proteins that do that job. They will pull this here, give it to DNA polymerase. For a small segment, it will add nucleotides. Again, it will let go of that. Then they will take another segment of DNA template and give it to DNA polymerase. This will add a small segment and give it back. This continues, okay? So from here, it takes it here, adds nucleotides, lets it go. Takes the next segment, adds nucleotides, lets it go. Because this has 5' end of the new strand here, it cannot add nucleotides at the 5' end. So on the other strand, synthesis happens discontinuous. So when I say 5' end to 3' end is the only direction in which DNA polymerase can function, I'm talking about the newly synthesized strand's polarity, not the template strand's polarity. If the template strand has the polarity 3' to 5' end, then on that strand, continuous synthesis will happen. And continuous synthesis will always happen towards the fork. Okay, because here only 3' end is there, here only it can add nucleotides newly. Like continuously it can add here. So if this is the direction of replication, if this is the direction of replication in this fork, over here continuous synthesis will be here. On this side, since direction of replication is there, continuous synthesis will be here. Okay, so if this side continuous synthesis is happening here, on the other side it will be the opposite. And here, if it is discontinuous, here it will be discontinuous. We are saying it is discontinuous, that does not mean it is slow. Okay, some people use the term leading strand and lagging strand. That does not mean on one strand replication happens very quickly, on the other strand it happens slowly. It pretty much happens at the same pace, but on one it is continuous, on the other it is discontinuous, because of our DNA polymerase's ability to extend the chain only in one direction, which is from 5' to 3'. Okay, so this is DNA dependent DNA polymerase's job. Now, whenever something new has to get added, whenever a bigger molecule has to form from smaller molecules, energy is required, right? So when you have something small, you're trying to build something big out of it, out of small molecules, you're building something, energy is required. You break something down, energy is released. Over here, building up is happening, smaller nucleotides are coming together to form a long chain. This requires energy. Where does the energy come from? So I'll tell you how this works, okay? So let's take this example again. I will erase this part. Okay, so let's say There is this DNA polymerase sitting here. Okay. Now, this is end. So, it will read that this has A. Opposite to that, it should add T. Right. So, we know that a nucleotide has a sugar, a nitrogenous base, and one phosphate. This is a nucleotide. But during replication, nucleotides containing a single phosphate are not used as the substrate. Nucleotides containing 3 phosphates are used as the substrate. These are called DNTPs, which means they are called deoxyribonucleoside triphosphates. The ATP that we talk about all the time is a DNTP. It has adenine here and 3 phosphates. Okay. Similarly, you have TTP, GTP, CTP and all of that. Okay. So what happens here is, this is T with 3 phosphates. It has 3 phosphates. Okay. The next nucleotide should be A. This will have 3 phosphates. We need only 1 phosphate. The 2 phosphates will break. Using that energy, a phosphodiester bond will form. Here we need only 1 phosphate. The other 2 phosphates are broken. Using that energy, the next phosphodiester bond is formed. Okay, so like that, these DNTPs perform 2 roles. Firstly, it is acting like a substrate for DNA polymerase. Secondly, it is also providing the necessary energy required for polymerization. Okay, so the same molecules, the same DNTPs, ATP, TTP, GTP, CTP, they are acting like the substrate during polymerization of this chain while simultaneously providing the energy that is essential for this process. So they are performing a dual role or dual function, they say. Okay, so that is about DNTPs. Now speaking of dependent DNA polymerases accuracy, I told you how this is about prokaryotes, and in prokaryotes like E. coli, once in every 20 minutes cell division is happening. If cell division is happening once in every 20 minutes, within 20 minutes, actually in around 18 minutes, DNA replication has to happen. Considering the amount of DNA that E. coli has, earlier we studied, right, 4.6 into 10 to the power of 6 base pairs, that's how long the DNA of E. coli is. If it has to replicate, then DNA polymerase adds 2000 bases per second. 2000 nucleotides are added to the chain per second. As we speak right now, as you're listening right now, this is happening inside of our bodies. Okay. DNA polymerases are, helping in replication and formation of newer molecules of DNA. And right now, very quickly, very rapidly, they're adding nucleotides to an extending chain of DNA. Okay. And it's important here, the speed is important, and along with the speed, accuracy is also important, because this is DNA. Any change in the nucleotide sequence is called mutation, and if mutation happens in coding regions, then it could cause any disorders, it could cause any abnormalities. So not only does the DNA dependent DNA polymerase have to be super quick, but they also have to make sure there is accuracy. You're not adding random nucleotides in between, right? So there are many repair mechanisms also inside the cells, where if at all there is any wrongly added nucleotide, if at all any mutation has happened, the cell will thoroughly check it and proofread, and all of that also happens. So all about that we will not learn in this chapter. So this is what we will learn. Now another question. On one strand there was continuous synthesis, on the other strand there was discontinuous synthesis, which means between this fragment and this fragment there is no phosphodiester bond. They are like separate fragments, but they are bonded to this strand by hydrogen bonds, but between these fragments there are no phosphodiester linkages formed. So in order to make sure that phosphodiester links form in between the existing fragments on the discontinuously synthesized strand, there is an enzyme called ligase. Ligation in general English means joining, right? So you would have learnt about ligase in biotech, where it's like the molecular glue and stuff. So this kind of helps form phosphodiester bonds between the existing fragments. These fragments have a name. It is not mentioned in your textbooks, but I will just let you know. These are called Okazaki fragments, named after a Japanese scientist. Okazaki fragments. Okay. Now, another thing which NCERT has just briefly mentioned, which is DNA dependent DNA polymerase cannot by itself start the synthesis of DNA. It requires an other molecule, they say. But actually what it requires is an RNA molecule. It is called an RNA primer. RNA primer first forms. After that, DNA polymerase continues the, you know, polymerization of the new DNA molecule. And then later that primer, RNA primer, will be replaced with DNA. This you do not have in your syllabus, but I'm just letting you know. Okay, so that's about the whole story of replication in DNA and how it happens in prokaryotes. In eukaryotes, it's a little bit more complex. In eukaryotes, since the genetic material, the DNA amount itself is so large, just one origin of replication will not suffice. So we will have hundreds to thousands of origins of replication, where simultaneously replication bubbles will form and replication will begin. More about that you will learn in your higher grades if at all you choose to pursue any degree in life sciences, then maybe you will study about that. For now, this is enough. If you've kept up until this point and you have no more confusions, then that is a win. Pat yourself on the back. So that we are done with replication, we will move over to transcription. Now, transcription, as we already know, is the process by which information that is present in DNA is copied onto a strand of RNA, right? I told you that the message in the nucleus has to get copied, get out of the nucleus, go to the ribosome and all of that drama. So the message getting copied from DNA to RNA is what we refer to as transcription. So here, it is a process where message is being copied. What is the message? It is the genetic information. From where is it getting copied? It's getting copied from DNA. What is it getting copied into? Into a molecule of RNA. Over here, we have to understand two differences between replication and transcription. In replication also, DNA got copied and formed another molecule of DNA. Here in transcription, DNA information is in DNA is getting copied to a molecule of RNA. Okay. Now, in replication, the entire length of DNA will get copied. So, how many ever chromosomes we have, how many ever genes we have, whatever is the amount of DNA that's present inside of our cells, all of that entirely will get copied, like an extra copy will get made. But here in transcription, that is not the case. Depending upon what information the cell requires at that point in time, only those segments will get copied. Okay. So it does not happen throughout the length of DNA. It happens only in specific segments. That's the first difference. Secondly, in replication, we had a double-stranded DNA molecule from which another double-stranded DNA molecule would form. Like from one, we would get two at the end of the process. So both of the strands in the template DNA in the parent DNA molecule would behave like the template. But over here in transcription, from a double-stranded DNA, we're getting a single-stranded RNA. So of the two strands, only one strand will act like the template, the other one will not. Okay. So when we are defining transcription, we define it as the process of copying genetic information from one strand of DNA into RNA. How does this function? How does this happen? What is the principle that governs this process? It's the exact same principle that governed the process of replication. Over there, DNA polymerase was the major polymerizing enzyme. It would look at the template strand and be like, okay, it has A here, let me add a T here, it has T here, let me add an A here. So based on the complementarity rule, it would add nucleotides on the new strand of DNA. Now, what happens in RNA synthesis or transcription also is the same. But here, since like I mentioned earlier, if this is like your DNA strand, let's say, okay, so you have ATGC here, you have TACG here. Let's say this is 3' to 5', this is 5' to 3'. Let's say this is the strand that is acting like the template. The new strand that forms will form from 5' to 3' end. This is not used as the template. So opposite to A, ideally, if it was DNA, T would be the complementary base. T would get added, but here we are talking about RNA. Does RNA have a T at all? It does not have a T, it has a U instead. So opposite A, complementary to A, instead of a T, you will have U. Everything else remains the same. Okay, so over here, adenosine forms base pair with uracil instead of thymine. Unlike replication, transcription happens only with a segment of DNA. Only one of the two strands is copied to RNA. All of these I've told you, right? Now let's talk about the actual process of transcription and understand why is it that only one strand of DNA has to act like the template. What would happen if both the strands behave like the template? What could go wrong? Like, why does it necessarily have to be one strand? There are two problems if both the strands of DNA behave like templates. Okay, what's the first problem, we'll talk about that. See, ultimately, if you look at the bigger picture, you have DNA. DNA is going to give rise to RNA. RNA is going to produce a protein, help in the synthesis of a protein. DNA is made up of nucleotides, RNA is made up of nucleotides, proteins are made up of amino acids. Okay. Different proteins have different sequence of amino acids. It's the same 20 amino acids, but in what sequence those amino acids are arranged varies from one protein to another protein. In our bodies, we have like thousands of proteins performing so many different functions. All of them are composed of the same 20 amino acids, but the difference lies in the sequence. Okay, so in what sequence those amino acids are present. Okay, so how does RNA know which protein to synthesize? All the proteins are getting synthesized in the RNA itself. How does it know right now I'm supposed to synthesize let's say a protein required in the plasma membrane? Right now I'm supposed to synthesize a protein required for the cytoskeletal element. How does the RNA get to know? I'm sorry, how does the ribosome get to know? The ribosome does the same job for all proteins. The sequence of amino acids is determined by the RNA. Based on the sequence of nucleotides in RNA, the sequence of amino acids in the protein will be determined. Okay, so RNA is actually carrying the message for synthesis of protein. Now, normally in one gene, one segment of DNA, there's information for a protein. Ideally, for information transfer to happen smoothly without any hiccups, if one segment of DNA codes for one protein, it will efficiently work. But in case both the strands of DNA are acting like the template, what could happen? Let's see. Here you have two strands. Assuming both these strands act like the template, let's say this one will give rise to this RNA sequence, this one will give rise to this RNA sequence. Now, if you look at this sequence and this sequence, they are very different from each other, right? Similar to how replication happened from 5' to 3', translation also will happen from 5' to 3'. Okay, so the mRNA is 5' to 3' and that sequence is what will determine the sequence of amino acids. Right now we have one segment of DNA. From this one segment of DNA itself, we are getting two different RNAs. And these two different RNAs are not, obviously, when I say different, they are different. They are not identical with respect to their sequence. Let's look at the sequence from 5' to 3' because this is how the translation process is going to proceed. Here 5' to 3' you have CUUGAC. Here 5' to 3' you have AGCGUC, which is different. If this two sequences are different, that means the proteins that you're going to get as a result of that is also going to be different. From one DNA segment alone, we will end up getting two different proteins. Overall, this will complicate the transfer of genetic information. Right now, one DNA, one segment of DNA is responsible for the formation of one kind of protein. So it's working wonderfully. But if the same segment contributes to the formation of two different proteins by producing two different RNAs, then obviously there will be like one circus inside the cell, right? So it will not be very efficient. So the whole information transfer thing will get complicated and confusing. So that's one reason. What's the second reason? Look at the sequence of this RNA molecule and this RNA molecule. If you look at the sequence, since they are formed complementary to these two strands will be complementary to each other. If you look at the 5' and here you have CUU, 3' and here GAA, this is complementary to this. So they will end up forming double-stranded RNA, and double-stranded RNA cannot synthesize proteins, right? So that way the entire exercise of producing RNA itself will be futile because protein itself is not going to get formed from it. Because of these two reasons, over the course of evolution, it has so happened that in a segment of DNA, only one of the strands will get transcribed so that transfer of information, genetic information, happens smoothly without any confusion. Okay, now we'll talk about the actual process of transcription. Like I mentioned earlier, if transcription has to happen, it's not like the entire length of DNA is getting copied to a strand of RNA. It happens only in specific regions depending upon the need of the cell at that moment in time. Now, if it's going to happen, supposed to happen in one segment, it's important that that segment is, clear. Okay, so there should be a clear starting point and a clear end point. This is where this segment begins. Here's where it's going to end. So transcription has to happen in this segment alone. I cannot go beyond the end side. I cannot start before the start side. So the enzyme should have clarity. There should be some sequences that are present at the end of the transcription side and the beginning of the transcription side, so that only that segment, information only in that segment will get copied, nothing additional is coming into your RNA. Okay. So, whichever segment of DNA is going to get transcribed, the information from whichever segment is going to get copied into an mRNA, that segment is called a transcription unit. Now, in this transcription unit, you can find three different components. You can find a start site, which we call the promoter. You can find an end site, which we call the terminator. What comes in between is called the structural gene. Over here, for representation schematically, this is the diagram that NCERT has given to you. But it's not like there's one dabba there for promoter, there's one box here for terminator. These are nothing but DNA nucleotide sequences. There is a specific sequence of nucleotides that kind of symbolizes that the start site is here. That signifies the start site. There is a sequence of nucleotides that will indicate the end site or the terminator site. They are all nucleotide sequences throughout. There is no line and box and nothing of that sorts there. So this is only for schematic representation. Okay, so this is the transcription unit. You have the start site of the promoter here, and then you have the end site of the terminator here, and then this entire segment is called the structural gene. This is the third component. Now, I told you there are two strands. Only one of them acts like the template. Whichever acts like the template is called the template strand. The one that doesn't act like the template is called the coding strand. Okay. If it is not even involved in transcription, what is the point of calling this the coding? What code is it giving away? What is it coding for? This is doing nothing. It's just sitting there, right? It's doing nothing. It's a transcription. It's the template strand that's doing the whole process, that's involved in the whole process. This guy doesn't even take part in transcription and we're calling it the coding strand. What is the reason for that? Let's try and understand. See, ultimately when protein synthesis is happening, where is the information coming from? It's coming from DNA. What is carrying the information? RNA. So it is the sequence of nucleotides in RNA that will ultimately determine the sequence of amino acids in a protein, right? Look at the sequence of the two DNA strands and this mRNA strand. If you look at the template DNA strand, compare it with RNA, you know that both of these are complementary, right? Because this is formed based on the principle of complementarity, these both are complementary. But if you look at the coding strand, this strand and RNA, look at the sequence, are they complementary? They're not complementary. They are similar in sequence, except in the RNA strand, wherever you have U, in the DNA strand, you will have a T, except for that every other nitrogenous base sequence is the same. It is this RNA that is ultimately going to determine the sequence of the protein. It is this RNA that is going to code for the protein. Similar code is present in DNA, one of the strands of DNA, even though it is not directly involved, directly participating in transcription. The sequence of nucleotides in both cases are similar. Then if this is coding for the protein and this also has a similar nucleotide sequence, it's not wrong in calling this strand the coding strand. Okay. So the strand, generally we say the strand that runs from 5 prime to 3 prime and is called the coding strand, and the strand that is from 3 prime to 5 prime is called template strand. If you have to locate the terminator region or the promoter region, we usually do that with respect to the coding strand. Upstream of coding strand, that means towards the 5 prime end of coding strand is where you will find the promoter. Towards the 3 prime downstream end of the coding strand is where you will find the terminator. Upstream means towards the 5 prime end. Downstream is towards the 3 prime end. Okay. So these are the different components that you can find in a transcription unit. Just like how we learnt about replication in prokaryotes, and then I told you about how it's different in eukaryotes. Even over here, we will learn about transcription in prokaryotes first, then I'll tell you about how it is different in eukaryotes. Okay. So we'll start off with transcription in prokaryotes. Okay. It's actually very simple. The enzyme that is involved in transcription is called DNA dependent RNA polymerase. Okay. Why is it RNA polymerase? Because what is getting synthesized now? RNA is getting synthesized. Adding more nucleotides to an existing RNA strand and making it longer is polymerizing it, right? So RNA polymerase is the enzyme. Why is it DNA dependent? Because this RNA polymerase cannot add nucleotides if there is no DNA template. It will see a DNA template, add complementary RNA nucleotides. So it needs a template to form a new strand. That's why we call it DNA dependent RNA polymerase. Previously in replication, it was DNA dependent DNA polymerase. Now it is DNA dependent RNA polymerase. So this guy, DNA dependent RNA polymerase, also can function only from 5' to 3' direction. So transcription proceeds in from the 5' to 3' direction, and this DNA dependent DNA polymerase sure knows to extend the chain of RNA, but it by itself cannot locate the start site or the end site. In order to identify and locate the start site or the promoter region, it requires the help of another protein factor called the sigma factor. When do we say transcription has begun? There are 3 steps in transcription: initiation, elongation, termination. When can we say it has started? When it has been initiated? You can say that when RNA polymerase, in this case represented by orange color, associates itself very briefly along with another protein factor called a sigma factor and binds to the promoter region. Here the promoter region I will represent in green, so you know the darker green. Okay, so this is where the promoter region is. So it's going to bind to that. Earlier in replication, I told you how there is an enzyme called helicase or unwindase that breaks the hydrogen bond, separates the 2 strands of DNA, and then replication proceeds. But here that's not the case. This RNA polymerase is a one-man show. It does everything. It only breaks the hydrogen bonds and continues to elongate the strand of RNA. So here this will bind to the promoter site. Once it is bound to the promoter site, you can say, okay, cool, transcription has begun now. It has it has gotten initiated. Soon after initiation has happened, this sigma factor will dissociate from the RNA polymerase. It's only a transient, very temporary kind of association that it has with the RNA polymerase. It will just get dissociated from that. Following that, RNA polymerase will continue to read the nucleotide sequence on the template strand, add corresponding complementary nucleotides and extend the RNA strand. Ultimately, when it is about to reach the terminator sequence, again, RNA polymerase is clueless. It does not know where to stop. So right now, it needs another friend to come and tell it like, dude, your job is done. Don't keep on going. It's over right now. It's time to stop. Your job is done, right? So for that, it requires another protein factor, which is called the rho factor. The rho factor is also called the terminator factor, or this is called initiator factor or whatever. So this is sigma factor. This is rho factor. So once the rho factor associates with RNA polymerase, they dissociate from the DNA. By then, the RNA would have completely gotten synthesized. Okay? Is that clear? I hope it is. Why am I asking you all like you can answer me right now? I sometimes forget that I'm recording and I talk to myself. Yeah. So that's what happens during transcription in prokaryotes. Now let's see what's different in eukaryotes. In eukaryotes, what happens is, if this is like the transcription site, not the transcription site, transcript unit, I'll tell. So if this is like a transcription unit, okay, so you have the 5' end here and you have the 3' end. At the 5' end, you have, wait. Let's assume this is the DNA. Let's think in DNA, you have promoter, you have 2 strands, right? You have promoter and you have terminator. And this portion here is called the structural gene. And within the structural gene, you will have certain regions which have information, and there are certain regions where information for protein synthesis is not present. The regions where information is present are called exons. They are expressing sequences. And the regions where information is not present, they are called introns because they are intervening. Intervening, coming in between the expressing sequences. Information is present only in the green regions, but these guys are present in between the green regions. They are intervening, so you call them introns. RNA polymerase, like I told you, is not very sharp. It cannot even identify where the promoter and terminator is, so it can't really make out that this is an intron, this is an exon. So whatever is present between intron and exon, it will end up copying. So in the RNA that is produced, you will have some necessary sequences along with unnecessary sequence. This RNA that is produced after transcription in eukaryotes is what we refer to as hnRNA, which is short for heterogeneous nuclear RNA. I'm talking about eukaryotes. In eukaryotes, transcription happens inside the nucleus. So over here, heterogeneous is because you have two different types of RNA here. You have non-coding sequences called introns and coding sequences called exons. You have both of them together within the same mRNA molecule. So you call that heterogeneous nuclear RNA. So all of this is happening where? It is happening inside the nucleus, right? So if you can think of this as a eukaryotic cell, I think I'm drawing this eukaryotic cell for the 400th time today, but yeah, it's okay. And this is our nucleus, this is our DNA, whatever. This is our RNA that has formed. Okay. So this RNA has to get to the ribosome to synthesize protein. But information is not there in the red regions. Before you push it out of the nucleus and send it to the ribosome, you have to ensure that the red regions are removed off and the green regions are put together, right? So that is one thing that needs to happen. Secondly, in the cytoplasm of eukaryotic cells, you have enzymes called exonucleases. These exonucleases, you know what they do? They start breaking phosphodiester bonds. Where are phosphodiester bonds present? They are present between nucleotides, adjacent nucleotides, right? So they will start breaking down one phosphodiester bond at a time from the end. Okay, so this side and this side at the 5' end and the 3' end. So in order to make sure that this ribosome successfully, produces a protein, you have to protect the two ends from this exonuclease activity. So additionally, at the 5' end and 3' end, they need protection. RNA needs protection. Otherwise, even before the protein is synthesized, RNA will get eaten up by these exonucleases. It will get broken down, right? So after transcription, three things have to happen. Introns remove, exons join together, 5' end has to be protected, 3' end also has to be protected. All of this will happen inside the nucleus itself before RNA leaves out and goes to the ribosome. So this process is called RNA processing, and this happens only in eukaryotes and not in prokaryotes. In prokaryotes, you do not have exons and introns, okay? Since eukaryotes have exons and introns, the genes are said to be split genes. Split because this is the actual gene, the green part, but it is split into multiple segments because of intervening sequences or introns. That's why you call them split genes. Okay, so now let's take a look at what are the different different modifications that happen. We'll start off with the first one, which is capping. Capping is specifically done at the 5' end where a modified G is added at the 5' end. How is it modified? It is methylated. Okay, so it is methyl guanosine triphosphate that is added at the 5' end. The exonucleases cannot recognize and bind to it. So that's end of DNA is protected from exonuclease activity. It cannot come and break bonds in between like endonucleases. They only work from the end, so that end is safe. It's also called G capping because it's a G cap that you're adding at the 5' end. What about the 3' end? At the 3' end, you have protection in the form of polyadenylation. What is polyadenylation? What is poly? Poly means many, many. Adenyl refers to adenosine residues like A, multiple of those A, as many as 200 to 300, will get added at the 3' end, a lot of them. Okay, and one important thing that you should know here is that when polyadenylation happens, it happens in a template independent manner. Earlier, how did the other nucleotides in RNA get formed? It was because of RNA polymerase, which was dependent on a DNA template. Over here, even without the template, there are enzymes that are capable of adding multiple A's at the 3' end in a template independent order. Okay, so that's one thing. What is the third thing? RNA splicing. Splicing is not the same thing as slicing. Splicing is more like joining, putting together. So splicing is where the introns are formed into loops and they are cut and detached, and all of the exon sequences, they will get joined together. Now this becomes functional mRNA. When it had intron and exon, it's not mRNA, it is called hnRNA or heterogeneous nuclear RNA. This heterogeneous nuclear RNA is a precursor of mRNA. Okay? So this is one change with respect to transcription in eukaryotes compared to prokaryotes. Another difference is that in RNA you have different types: mRNA, rRNA, tRNA, snRNA, all of these. Even in rRNA you have multiple types. Okay. In prokaryotes, there's just one type of RNA polymerase which will transcribe all different types of RNA. But in eukaryotes, you have three different types of RNA polymerases. You have RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. How do you remember which one transcribes what? You can remember it with the help of this one word, remote. Okay. So in the word remote, R comes first, M comes second, T comes third. That is how it is. RNA polymerase 1 will transcribe three different types of rRNAs. Which are the three different types of rRNAs that it will transcribe? 28S rRNA, 18S rRNA, 5.8S rRNA. All of them have an 8 in them. 28, 18, 5.8. mRNA. See, these are eukaryotes. Direct transcription will not happen of mRNA. First hnRNA will form, then only mRNA will form. So even though it's saying mRNA here, RNA polymerase 2 transcribes the precursor of mRNA, which is hnRNA. This is the precursor of mRNA. So that gets transcribed with RNA polymerase 2. RNA polymerase 3 will transcribe three different types of RNAs and also tRNA. So it transcribes tRNA, one type of ribosomal RNA which is of 5S type here, and it will transcribe small nuclear RNAs that are formed and present inside the nucleus. If you're calling them nuclear RNA, obviously it's present inside the nucleus. If you're calling them small, obviously they're going to be they're going to be short in its length. Okay, so this is tRNA, 5S rRNA, snRNA. So in eukaryotes, you find three different types. In prokaryotes, you find only one type. In eukaryotes, it's found inside the nucleus because that's where transcription takes place, right? Yeah. So that's about transcription and how it is different in prokaryotes and eukaryotes. Now let's talk about gene versus cistron. So if this is like our DNA, again drawing it for the 7964th time today, this is our, oh, did you see how we got a heart here? Okay, so this is DNA. In different regions of DNA, you have information for different things, different types of RNA. Let's say this region has information for the synthesis of a type of ribosomal RNA. This region has information for the synthesis of a type of tRNA. This region has information for the synthesis of a type of mRNA, of course via hnRNA. And this region has information for the synthesis of small nuclear RNA. Okay. All of these genes that are transcribing rRNA, tRNA, mRNA, snRNA, any type of RNA. If DNA segment has information for the synthesis of a type of RNA, then you call all of these cistrons. I'm sorry, you call all of them genes. Okay, so this segment is also a gene, this segment is also a gene, this is also a gene, this is also a gene. If it has information for the synthesis of an mRNA, you call it a gene. But in order for you to call a gene a cistron, it should be producing a protein or a polypeptide via an mRNA. Only genes that produce mRNA and via mRNA produce protein, only such genes are called cistrons. If it has information for any type of RNA, it is a gene. If it is a gene producing mRNA through which protein is getting synthesized, then you call that gene a cistron. All cistrons are genes, but all genes are not cistrons. Okay. So here, the gene is defined as the functional unit of inheritance. The DNA sequence coding for rRNA, tRNA are also defined as a gene. Cistron is a segment of DNA that specifically codes for a polypeptide or what we call a protein. Okay, so that is gene versus cistron. Let's talk about the difference between prokaryotic transcription and eukaryotic transcription. Prokaryotic transcription happens in the cytoplasm. In eukaryotes, it happens inside the nucleus. In prokaryotes, you have just one type of RNA polymerase. In eukaryotes, you have three different types. In prokaryotes, what happens is the RNA that is formed is polycistronic, which means if this is like an RNA molecule that is formed, within this single RNA molecule, there will be information for three different proteins. Not necessarily three, I mean many different proteins. Let me just write more than one protein. Okay. In eukaryotes, if there is one RNA formed, this will code for only one protein. After transcription, there is no further processing required in prokaryotes, but in eukaryotes, processing is essential. Next, we'll talk about the genetic code. Well, as the name itself indicates, genetic code is a code, which means it is a coded message, right? So what is the coded message? You remember back in the day when we were little, when I was probably your age, they used to exchange letters, right? Secret coded letters used to be exchanged where A is equal to 1, B is equal to 2, C is equal to 3. If somebody has to call you bad, let's say you have to use the word bad, instead of using the word bad as it is, B being the second letter would be 2, A, 1, D, 4. So this reads as bad, okay. So this sort of coded language used to be exchanged between people if they didn't want people to understand what it is about, okay. So that's a very simple example that I'm giving to you right now. Where is this coded message essential? It is essential during translation. So, I mean, it's not like essential. Where does this happen? It happens during translation. See, during translation, what's going to happen? The sequence of nucleotides in RNA is going to determine the sequence of amino acids in the protein or the polypeptide, right? So, RNA is speaking the language of nucleotides, protein is speaking the language of amino acids. It's like the numbers and letter kind of code that we spoke about right now. So, how exactly can we say that this particular nucleotide is responsible for this amino acid? If there is this nucleotide sequence, it's responsible for another amino acid. What is that code exactly? We know 2, 1, 4 is bad because I already know that A is 1, B is 2, C is 3. If I know the code, it will be easier for me to understand what is written in the letter in the form of numbers. If I do not know the code, I will not know what sequence of nucleotides will give me what sequence of amino acids. So a lot of people were researching about what exactly could this code be, and yeah, many of them contributed in deciphering the genetic code, and that's what we're going to learn about today. We'll start off with George Gamo's contribution. So basically, as we already know, the number of different nucleotides that you have in RNA is 4. You have AUGC, right? 4 different nitrogenous bases, as a result of which you have 4 different nucleotides. But in proteins, you have how many amino acids? 20 amino acids. So if it was like one nucleotide or one nitrogenous base codes for one amino acid, we would have enough codes only for 4 amino acids. What about the remaining 16 amino acids? What would their codes be? That's not possible, right? So it's not possible that one nucleotide will code for one amino acid. Then could it be a combination of 2 bases, like AU codes for one amino acid, GC codes for another amino acid, GA codes for another amino acid? If you apply all the permutation and a combination formulae that you know, you will know that for 4 nucleotides, the maximum number of combinations you can get where there are 2 nucleotides in one particular code is 4 to the power of 2, which is 16. However, we are still less than 20 amino acids. What about the remaining 4 amino acids? What would their codes be? So George Gamow suggested that it's likely that the genetic code is a triplet. Possibly every 3 nucleotides, consecutive nucleotides, is going to produce or code for one amino acid. Okay, not produce, code for. That's the right term. Okay. So that is more feasible, more likely, because if it is a triplet code, then 4 to the power of 3, we have 4 times 16, 16 4 times 64, a total of 64 different combinations of nucleotides, AUG, GUG, CUC, AAA, UUU, like that if you think about, you have 64 different combinations that arise when the codon is triplet, even though there are just 4 nucleotides. So that is far higher than the 20 amino acids that we have, right? So if it is a triplet codon, it's possible for every amino acid to have at least one code. So he suggested that a code determining one amino acid is made up of 3 nucleotides, and that would generate a total of 64 codons. Codon is nothing but another term for code. Think about it like that. Just like how I said A is equal to 1, think about how 3 different nucleotides consecutive, like next to each other, right next to each other, they 3 will determine or code for one amino acid. Another 3 nucleotides will code for another amino acid. So for one amino acid, information is present in 3 nucleotides, 3 immediately adjacent nucleosides. Okay, 3 consecutive nucleotides will give code for one amino acid. Okay, that's what he said. And then our Indian researcher Hargobind Khurana, he figured out a way to synthesize RNA molecules in combinations that we require. Okay, it's not like we're synthesizing RNA using a DNA template. In whatever sequence you want RNA molecules, if you want an RNA molecule that has only U, if you want an RNA molecule that has only C, you want an RNA molecule that has CU, 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 whatever. Okay, so based on your requirement, you can synthesize artificially in the lab RNA molecules. Okay, so he synthesized RNA molecules with defined combinations of bases, whichever ones he requires. And there's an enzyme which was, discovered much before all of this happened. It was already known. They use that enzyme also in deciphering the genetic code, and that enzyme is called Severo-Ochoa enzyme. Severo-Ochoa is the name of a scientist. It's also called polynucleotide phosphorylase. I'll tell you what is the advantage of this enzyme. This enzyme was helpful in polymerizing RNA with defined sequences. It could synthesize RNA even in the absence of a template. Okay, so this is the enzyme that Hargobind Khurana used in order to synthesize RNA nucleotides of a sequence of his choice. Right now, what is the problem for me? I know that DNA is going to get copied as RNA, RNA is going to get out of the nucleus, go to the ribosome. In the ribosome, reading the message on RNA, polypeptide is going to synthesize, but I do not know which sequence of nucleotides or which codon will give rise to code for which amino acid, which sequence codes for which amino acid. That's what we're trying to figure out here. So I'm going to synthesize, not me, like Hargobind Khurana. He synthesized RNA molecules of his choice. The sequence was of his choice. And he synthesized them using Severo-Ochoa enzyme. And then there was this other guy called Marshall Nirenberg. What he did was, he started synthesizing proteins in cell-free mediums. If protein synthesis happens inside of our cells, inside of living organisms, it happens always inside the ribosome itself. It's a process that happens like within the cell of living organisms, right? So what he did was, even in the absence of cell, in vitro, like in the laboratory conditions, he could synthesize proteins. Okay. So yeah, he used a cell-free system for protein synthesis that finally helped the code to be deciphered. So what are we doing now? George Gamow suggested every code has 3 nucleotides. Okay, fine, we got it. Which 3 nucleotides codes for which amino acid is still a question mark. We are trying to figure that out. So what did Hargobind Khorana do? He used Severo-Ochoa enzymes and generated sequences of nucleotides of RNA. Like he synthesized RNA molecules and the sequence of nucleotides was of his own choice. He knows that in this RNA molecule, UUU is present throughout. He knows that in this particular RNA molecule, there's A throughout. Okay, so only A is present. So based on his requirement, he synthesized RNA molecules of defined sequences. These sequences he already is aware of. Okay, and then Marshall Nirenberg started synthesizing proteins in the lab. Whatever is required for protein synthesis, you provide all of those requirements, all of the enzymes and all of the structures required in a laboratory setup. So in vitro, even outside the cell, proteins are being synthesized using the RNA that Hargobind Khorana has synthesized. Okay, so you have the RNA, you synthesized a protein in the lab, in vitro. You analyze the sequence of amino acids. You already know the sequence of nucleotides. So based on what sequence of amino acids you've got in your protein, you can determine what is the code, right? If the first 3 nucleotides in the RNA that you have synthesized is UUU, in the corresponding protein of the first amino acid is phenylalanine, you know outrightly right there that UUU codes for phenylalanine, right? So somewhere else you have GUG that codes for valine, somewhere else you have AUG that codes for methionine. So it is easier right now to find out which 3 nucleotides code for which amino acids, okay? So that's what we're talking about here. So ultimately, after using different sequences of nucleotides of RNA, as you can see here, they first synthesized what we call copolymers. Copolymers will have nucleotides of the same kind repeating multiple times. Like all you have here is U. If you had U here, see here, this is 1 codon, 2 codon, 3 codons, 4th codon, 5th codon. You get amino acids that have, you get a polypeptide chain that has 5 amino acids. Totally you have 15 nucleotides. From that you're getting 5 amino acids. Everywhere there is UUU, so all the amino acids in this particular polypeptide chain are going to be phenylalanine. From this we got to know that phenylalanine is coded by UUU, that is the codon for phenylalanine. Like that they do it with AAA, CCC, GGG, everything else and find out. And they will also, they also tried to synthesize what we call heteropolymers, where the RNA is not just made up of one nucleotide like this, it is made up of a mixture of 2 or more nucleotides. In this case, this is a heteropolymer that contains U and C alternating with each other. From this, you can get to know 2 codons. One codon for UCU, which is serine, CUC, leucine. So from this, we got to know the codon for 2 amino acids. UUU, phenylalanine, we know. So 3 amino acids code, we know. Then we will try it with different sequence of nucleotides and eventually figure out which 3 nucleotides code for which amino acid. And ultimately, they could figure out the entire genetic code. And this is the entire table that will give you which 3 nucleotides code for which amino acid. Okay, so yeah. We'll talk about the features of genetic code. Firstly, we already know this. The genetic code is a triplet, which means 3 nucleotides will code for 1 amino acid. You have a total of 64 combinations possible using 4 nucleotides present in RNA. So totally 64 are possible, but all of them will not code for proteins. All of them will not code for amino acids. There are 3 of those 64 which will not code for any amino acid. They are called the stop codons. See, UAA, UAG, UGA, these are the stop codons. You can remember it like this. You are away, UAA. You go away, UGA. You are gone, UAG. Okay, so you are away, you go away, you are gone. These are the 3 nucleotide sequences or the codons that will not code for anything, they are called stop codons. Okay, so out of the 64, how many are functional? Only 61 are functional. The other 3 do not code for any amino acid. What is our next step? Next point. Some amino acids are coded for by more than one codon. Of course, you can see that a lot here. You can see how leucine is coded for by 6 codons. All of these 6 codons will code for leucine. And you have serine that has 6 codons. There are 4 here and 2 here. Then you have arginine that has 6 codons, 4 here, 2 here. Glycine here has 4 codons, lysine here has 2 codons, proline has 4 codons. So it's possible that certain amino acids have more than one codon. If one amino acid has more than one codon, then you call it degenerate. It's possible, right? There are 61 functional codons, there are only 20 amino acids. It's possible that one amino acid can have more than one codon. But is it necessary that every amino acid will have more than one codon? Not really. If you look at methionine here, it is only one codon. Here, methionine has only one codon. Nowhere else you will find another codon for methionine. So it's not mandatory that every amino acid will have more than one codon. However, there are a lot of amino acids that are coded for by more than one codon. Now, that's degeneracy. Next step. The codon is read in mRNA in a contiguous fashion. There are no punctuations. What does this mean? It means that whenever you have nucleotide sequences like A, U, G, C, 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 U, A, G, something like this, if these 3 are specifying one nucleotide, one amino acid, then the next codon will not be here. There is no skipping of nucleotides in between. Every nucleotide is part of one codon. So this is one codon, this is one codon, this is one codon. There are no pauses, there is no skipping of nucleotides in between. Every nucleotide in mRNA will be part of a codon and it will be part of one codon only. You can't find overlapping also here. Non-overlapping is also a property of genetic code. Non-overlapping basically means if this is one codon and this is the second codon, the third codon cannot start from here. It can't be that this C is part of this codon as well as this codon. Every nucleotide in RNA is part of one codon. It is part of one codon only. It cannot be skipped. Always it will be part of a codon and only one codon. Okay. So, there is no skipping, no punctuations in between. So, that's what the second or third point means. Fourth point, it is nearly universal. Universal means, you know, right? It applies everywhere. That is what it means. So here we are calling it nearly universal because obviously, like most rules, this also has an exception. In a vast majority of cases, if UUU codes for phenylalanine in one organism, in another organism also it will code for UUU. This is what we use as a property for RDNA technology, right? We can make bacteria produce our insulin because in our insulin producing gene, whatever nucleotides are there, whatever insulin gets produced, if that gene is put inside a bacteria, it follows the same genetic code. So it is possible for us to obtain our proteins by using other organisms. So that is the basis for RDNA technology. But it so happens that in some cases, like in certain genes in mitochondria and in certain protozoans, the genetic code is slightly different. So that's why we call it nearly universal and not outrightly universal. Next, AUG has dual functions. I told you how AUG has only one codon for methionine, right? I told you here methionine has only one codon, that is AUG. This is the start codon. I told you how these 3 are called stop codons because they do not code for any amino acid. This is called a start codon. That signifies the start site for translation. AUG will code for methionine. Not only does it code for methionine, it also acts like a start codon. That is why it's performing two functions. That's why we say AUG performs dual functions. Okay, so one, it codes for methionine, and another, it acts like a start codon or an initiator codon. These are some of the salient features of the genetic code. Now let's talk about mutations. We already know what mutations are. Very briefly, we've touched upon it in the chapter called Principles of Inheritance and Variation, right? We've spoken about the sickle cell anemia disease. I told you how there is a substitution mutation in the 6th position of the beta globin chain of hemoglobin, right? We've learnt about that. So here we're going to, deal with it in a little bit more detail. So yeah, mutations are of many types. You have point mutation and you have gross mutation. Point mutation is when it happens at a single nucleotide or a single base pair level. Gross mutation is when it is more than a single base pair or nucleotide level. In your syllabus, you have to learn about 3 different types of point mutations. One is a substitution mutation, insertion mutation, and deletion mutation. Substitution means what? What is a substitute teacher? One day in school, your English teacher did not come, she was on leave, and then they send a substitute teacher who probably teaches Hindi in some other section. So it's like replacement, right? Whenever you have a PE class in school and then the math teacher hasn't finished their syllabus, they will come and take the PE class, right? So it is substitution means replacement. So here, instead of one base pair, another base pair is replaced. Then you call it substitution mutation. Insertion means within an existing segment of DNA, you insert another base pair which already did not exist there. A new base pair somehow pushed in between the existing DNA molecule. Deletion, as the name itself indicates, one base pair removed from an existing DNA molecule, that is called deletion. What are the consequences of these mutations? We'll try and understand. So first we'll talk about substitution. Here are 2 sequences of DNA. So this is the DNA. There is DNA, mRNA, amino acids, all of them given here. This is the normal DNA, template strand. This is mRNA strand. This is the polypeptide strand. Okay, so here you have the template DNA. So let's say at this position in the template DNA strand, normally C used to be there. But right now, C, because of substitution mutation, is replaced with T. As a consequence of that, in the mRNA also, instead of G, you have an A, right? RNA polymerase does not know if any mutation has happened, should it fix it? None of that RNA polymerase is aware of. Based on what is there in the template, it will just end up synthesizing a new strand of RNA, right? I told you that every 3 nucleotides are responsible for coding one amino acid. So here the code is changed from here to here. Here it was GAA, it coded for glutamic acid. AAA codes for lysine. So in whichever codon substitution has happened, that codon's amino acid may be changed. It's not like every time it will be changed, because sometimes, as you can see here, the same amino acid can be coded for by many codons. Let's say some substitution mutation happened and in the codon, instead of CUU, CUC is there. Even if substitution mutation happened, you will not get a different amino acid because this amino acid is coded for by both of these codons, right? So it's not like every time there is a substitution mutation, whichever codon is involved, that has to produce or code for another amino acid. It may happen, it may not happen, but it could happen. Okay, so if the codon that has been modified after substitution is coding for the same amino acid, great, no problem. But if it's coding for a different amino acid, then in that sequence of amino acids, in that position, you can find a change in the amino acid sequence. This is far less harmless compared to insertion and deletion, because here when substitution happens, whichever codon is involved, only that amino acid will be changed. No amino acids before or after that will be altered. Everything else beyond this will still remain the same because it has only gotten replaced. None of the other codons beyond this site of mutation have been altered. But if you look at what happens in insertion, it's a little different. So here again you have the template DNA strand, mRNA and amino acids. So here insertion has happened at this position. In normal DNA, the third nucleotide sequence, like the third codon, was CTT. But in between this G and this C, another C has been inserted here. What happens as a result? Every codon from this point onwards will be altered. Whatever was part of this codon initially will now be part of this codon. Whatever was part of this codon initially will now be part of this codon. It's almost as if the entire reading frame has shifted to the right. What was here will move on next. What was here will move on to the next codon. So it has kind of shifted to the right. So in such cases, from the point of mutation, from the point of insertion, every other nucleotide will be altered. the sequence of every other nucleotide will be altered, and such a mutation is called a frame shift mutation. See, whenever you're answering an exam using an OMR sheet, you know that if you make a mistake in one question, let's say you're answering like 10 questions and the 7th question, you answered one question incorrectly. The answer was supposed to be C, but you marked it as D. So only that answer will be wrong. Let's say you're answering or bubbling the OMR sheet, and it so happens that at question number 3, instead of bubbling the answer for question number 3, you end up answering bubbling the answer for question number 2 one more time. What happens after that? What was supposed to be 3 will now be 4. What was supposed to be 4 will now be 5. From that point onwards, every answer is going to be incorrect. Right? Same thing happens in a frame shift mutation. Okay, so whenever insertion happens, it will lead to a frame shift mutation. Whenever deletion happens also, it will lead to a frame shift mutation. Deletion is like, over here you see ATG was the second codon, the second 3 consecutive nucleotides. Here, the A from here has gotten deleted. So whatever was a part of this codon has now moved to this codon. What was initially part of this codon has now moved to this codon, and the entire reading frame has kind of shifted to the left. So even over here, from the point of mutation, you can notice how every amino acid afterwards, every codon afterwards will be altered. And this also is a frame shift mutation. Whenever insertion and deletion happens, that will result in frame shift mutation. However, if there is an insertion and deletion of nucleotides that are multiples of 3, then there won't be a frame shift mutation. Okay, so if you introduce 3 nucleotides, let's say, instead of this was what? Deletion. This was, deletion. Deletion or insertion. If you delete, let's say, instead of, let me just copy this one more time so that's easier to explain. Yeah, I'll copy the slide. So what happens? Instead of, so let's say instead of this one nucleotide getting deleted, let's say these 3 nucleotides got deleted. Okay. So what happens in that case? Only this codon and this codon will be affected. This will become part of one codon, everything else will remain the same. Okay. So again, same thing, if these 3 were deleted, this and this will become one codon and the rest will remain the same. If these 3 were deleted, just this codon will be affected, the rest will be the same. If it is insertion or deletion of nucleotides in 3 or multiples of 3, then frame shift won't happen. If deletion of insertion of nucleotides or base pairs are happening in numbers that are not multiples of 3, then frame shift mutation will take place. Okay, yeah. So now that we're done with mutation, we will talk about translation. We will actually get into the most important part, translation, how proteins get synthesized. This is such a beautiful process. You will love it. Okay. So for the synthesis of proteins, we require 3 different types of RNA. mRNA, obviously, that's what is carrying the message. You need rRNA. Where is this rRNA? rRNA is present inside the ribosome. Where does protein synthesis happen? Protein synthesis happens inside the ribosome, right? So ribosome is made up of rRNA. If you recollect what we studied in Cell the Unit of Life, I told you how ribosome is not like other organelles where there is a membrane around it. It is a non-membrane bound organelle. If it's not a membrane bound organelle, then what is its structure like? Ribosomes are only made up of 2 different things. One, it is made up of different types of RNA, which we call ribosomal RNA, and it is associated with some proteins. Okay, so just ribosomal RNA and proteins will build a ribosome, the organelle. So the ribosome, inside that it's where protein synthesis is happening. So rRNA is involved. If ribosome is involved, ribosome has rRNA, so rRNA is involved. It is a structural component of the ribosome, right? That is what ribosome is made up of. Apart from that, the RNA present in the ribosome also catalyzes peptide bond formation. Now, what is a peptide bond bond? Peptide bond is the bond that links 2 amino acids together in a long chain. When we discussed how a polynucleotide chain forms, I told you how 2 nucleotides are joined together by means of a phosphodiester bond. Right now, we're talking about protein synthesis, and in proteins, amino acids have to be joined together to form a long chain. And in order to link 2 amino acids together, a peptide bond will form. And for the catalysis of that peptide bond, rRNA, which is component of the ribosome, is going to get involved. Okay, so that is what will act like the enzyme there. What about the other type of RNA called tRNA? I mentioned earlier that for uh, translation to happen, you need a translator that understands both languages. Over here, translation needs to happen. Nucleotide language has to be understood and communicated so that the protein forms, right? So, this tRNA molecule can understand nucleotide language, obviously, because that is also a nucleotide sequence, that is also a type of RNA. And it also understands the polypeptide language because it understands the amino acid language. Okay. So this tRNA molecule acts like the translator. Not only does it act like the translator for protein synthesis, amino acids are required. Protein synthesis happens in the ribosome. Free amino acids are present in the cytoplasm. So this tRNA carries the amino acids present in the cytoplasm, takes it inside the ribosome, reads the code and makes sure that the amino acids are linked in the correct sequence. Okay, so for that you require three different types of RNA. mRNA carrying the message, so you call it the messenger RNA. tRNA is called transfer RNA, that not only brings amino acids, it also reads the genetic code. rRNA, the RNA present in the ribosome, this plays the structural role as well as catalytic role. Structural role, why? Because it is a structural component of ribosome. Catalytic role, because it is what catalyzes the peptide bond formation between two adjacent amino acids. Now, let's talk about tRNA molecule because we haven't spoken much about it. So what is a tRNA molecule? That is also an RNA like mRNA, hnRNA, rRNA. tRNA is also a type of RNA. It's a single-stranded RNA, but it forms double-stranded regions by folding upon itself. Okay. So I told you that this tRNA can speak nucleotide language and also speak amino acid language, right? So in this tRNA molecule, you can find an amino acid acceptor end. And this amino acid acceptor end will specifically be present at the 3' end. At the 3' end, it will have an amino acid acceptor site, and it has an anticodon loop over here in between. What is an anticodon? Something that is complementary to a codon. Where do you find a codon? In an mRNA. If in mRNA, AUG codes for methionine. Okay. Okay, so here they've taken AGU as example. Let's use the same. Here you can see that on the mRNA, there is a codon AGU. We know that AGU codes for the amino acid serine. But this tRNA is what is going to get serine from the outside in the cytoplasm into the ribosome. Okay. How it binds with the mRNA while protein synthesis is by means of what we call an anticodon. The anticodon will have a complementary sequence to codon. If codon in mRNA is 5' AGU 3', anticodon in tRNA will be 3' UCA 5'. Corresponding to this, there will be an amino acid attached at its 3' end. Okay. So, yeah. You can see even over here, this is the 3' end, 3', this will be 3', 3' UCA 5'. That is the anticodon sequence. They complementarily bind with each other. Okay. Yeah. So it has an anticodon loop and it has an amino acid acceptor end as well. For starting translation, there is a specific initiator tRNA that carries methionine, the initiator amino acid. Okay. For the stop codons UAA, UAG, UGA, there are no tRNAs. In its secondary structure, this tRNA looks like a clover leaf. And in its actual three-dimensional structure, it appears like an inverted L. This is normal L, this is inverted L. Right? Ulta 7 or ulta L, that sort of a shape it will have. Okay? Yeah. So now let's talk about translation. Translation is nothing but protein being synthesized. For protein to get synthesized, energy is essential because we are taking tiny amino acids and putting together and making a long chain. So it requires energy. Anything bigger, if it is being built using smaller components, that will consume energy, right? I told you how during DNA synthesis energy is consumed, during RNA synthesis energy is consumed. In that case, energy was provided by the DNTPs themselves. They acted as the substrates as well as provided energy required for the phosphodiester bond formation. But over here, that is not the case. During translation, amino acids have to be charged so that, tRNA has to be charged so that energy is provided for protein synthesis. Okay. So first step that happens in translation process is the activation of amino acids. Activation of amino acids is when amino acids react with high energy molecules called ATP, you know about them, right? And form what is called amino acyl AMP, adenosine monophosphate. Adenosine triphosphate has 3 phosphates. This has clinged on to adenosine with 1 phosphate, adenosine monophosphate, and the remaining 2 phosphates are released as pyrophosphate. We represent it as PPI. Now, this amino acid which is attached to an AMP is called activated amino acid. Now, this activated amino acid has to get attached to the tRNA at its 3' end. That process is called amino acylation of tRNA or charging of tRNA. So, this amino acyl AMP, amino acid along with AMP, combines with tRNA. This is looking like a trishula. This is just showing the anticodon end here and the amino acid acceptor end here. So, to the amino acid acceptor end here, this activated amino acid will come and bind and the AMP molecule will get released. Now, this guy is fully charged. It is now capable of bringing about translation and synthesizing proteins. Even without the ribosomes, if 2 charged tRNAs come close to each other, they have enough energy to form peptide bonds, but still inside the RNA, it is getting catalyzed and happens at a much faster rate. So, protein synthesis happens inside the ribosomes. Now, we'll start off with initiation. So, first we have this mRNA strand that has been transcribed. Okay. Translation proceeds from the 5' direction to 3' direction in this way. To this mRNA strand, the small subunit of ribosome will first come and bind where? At the start site. In the mRNA, you have 5' end a G cap, 3' end you have the poly A tail, and here at the 5' you have the start codon, and at the 3' end you have one of the 3 stop codons, in this case UAA. Okay, so to this mRNA strand, the small subunit will first come and bind, and then to this complex at the start codon, the initiator tRNA carrying methionine as the amino acid at its 3' end will come and bind here. That's the next step. Next to this complex, the large subunit of ribosome will come and bind. You know that ribosome exists in the form of 2 subunits, a large subunit and small subunit. They can exist separately inside a cell, but whenever protein synthesis needs to happen, they both will come together. Okay. So now it is ready. Okay, initiation has happened perfectly. Everything is aligned for translation to take place. Within the large subunit of the ribosome, there are 3 sites. Okay, one is called the P site, one is called the A site, and one is called the E site. A site is acceptor site, P site is peptidyl site, and E site is exit site. So, to the P site, at the P site, the first tRNA, the initiator tRNA carrying methionine will come and bind. Right now, A site is empty. It is vacant. Corresponding to whatever codon is present next, the second codon, the tRNA carrying a corresponding anticodon and amino acid, this charged tRNA is going to enter the large subunit and come and bind to this second codon over here. Okay, so this is going to happen. Now, this second codon is UUU, anticodon will be AAA. UUU codes for phenylalanine. So, this tRNA is carrying phenylalanine as 3' end. So, this is going to come here and bind here. Okay. So, when these two are brought close to each other, peptide bond formation can happen between phenylalanine and methionine, but still it is catalyzed by an enzyme called 23S rRNA, which is present in the large subunit of prokaryotic ribosome. Okay. So, here within the large subunit, you have a type of RNA. Of course, large subunit, small subunit, they are made up of rRNA. There is one type of rRNA called 23S rRNA in prokaryotes that will bring about the formation of peptide bond between this amino acid and this amino acid. After that, the bond or this tRNA will get detached from this amino acid and leave out of the E site, which is the exit site. It'll get out. Okay. Next, what happens is elongation. The ribosome will start sliding over mRNA, reading one codon at a time. So as the ribosome slides over the mRNA, what was in A site will now be in P site. A site will be vacant. Okay. So right now UUU is in A site. Once ribosome moves here, what was in P site, A site will now be in P site, AA is in P site, and the next site, A site, will be vacant. So the next corresponding tRNA will come and bind here. rRNA 23S rRNA will catalyze the peptide bond formation, and this will keep on continuing till it reaches the stop codon. Once it reaches the stop codon, this is not elongation, this has to be termination. This is the last step in translation. This is called termination. As we already know, there are no stop codons for, there are no tRNAs for stop codons. Since there is no tRNA for stop codon, another protein factor called release factor will come and bind at the A site whenever the ribosome reaches the stop codon. Once the release factor comes and binds here, all of these will get dissociated and ultimately the protein synthesis will be completed. So that is what happens during translation. Now we have to learn about something called untranslated regions. What are these untranslated regions? Untranslated regions are regions that are found within mRNA. They are found after 5' and before start codon, after stop codon, before the poly-A tail. Okay, so after G cap, before start codon, in between the G cap and the start codon, in between the poly-A tail and the stop codon. Okay, so did I say stop codon here? I can't recollect. Okay, so I meant start codon. So these regions are called untranslated regions or UTRs. These are present in mature eukaryotic RNA. They don't directly take part in translation. They are not translated. They are not codons that will specify amino acids, but they will make sure that the translation process happens efficiently. Okay. Now, difference between transcription, translation in prokaryotes and eukaryotes. In prokaryotes, transcription, translation, both of that happens in the cytoplasm only because there is no well-defined nuclear membrane. So, transcription and translation can be coupled. After transcription, mRNA will form. I've represented that in green color. This ribosome first started to translate RNA. From here to here, it has reached here and it's formed a long polypeptide chain. To the same mRNA, many ribosomes could simultaneously be attached and start translating it. So this forms a structure that is called a polysome. You can find this in prokaryotes, not in eukaryotes. Because in eukaryotes, there is spatial separation between where transcription happens and where translation happens because of the presence of a membrane-bound nucleus. Within the nucleus, transcription happens. Outside the nucleus, translation happens in the ribosome. So it cannot be coupled. It cannot happen together. Or many ribosomes do not read the mRNA at the same time in eukaryotes. That also. Polysomes don't form in eukaryotes. Next. Yeah, now let's talk about regulation of gene expression. I've told you how not all gene products are required by a cell at all times, right? So when we're growing, when we form, like how did our life first begin? We began life as a zygote, right? When the sperm and the egg meets, a zygote forms. That's the first cell of multicellular organisms. And that will have the complete set of genetic information. It will have all the 24 chromosomes. But as and when the zygote develops and forms the embryo, one cell divides to give rise to 2, 2 becomes 4, 4 becomes 8, and so on. The number of blastomeres will go on increasing. And once gastrulation happens and the germ layers are laid, every cell knows what it is going to become a part of in the future. Some cells in the ectoderm will know that is going to become part of the skin. Some cells in the mesoderm probably know they're going to become part of the heart or whatever. So once they know what they are going to become in the future, they will, in those cells, some genes will be permanently switched off. Like for example, you will never see that the cells in our heart will secrete neurotransmitter, or the cells in our brain will secrete some enzyme that your gut secretes. So that does not happen. Wherever whichever product is not required, those genes will be permanently closed, turned off, we would say. And in other cells, like where genes are actively being transcribed, it's not necessary that every gene product is required by the cell at all times. So sometimes it may be kept on, sometimes if it's not necessary, it is turned off, right? Why do genes have to be turned off and on? That's because transcription, translation, replication, all of these are energy consuming processes. They require energy. If you do not want a product, what is the point of spending so much energy in producing it? Right? If that protein is not essential by the cell, just because you have information for that, you shouldn't be, you know, producing it. Just because you have electricity in your house, you don't turn off the switches of your light and fan and refrigerator even when you're on a vacation, right? So when it is necessary, only then you will turn it on. When it's not used during winters, cold winters, nobody puts your AC or um, fan on unless you're a psychopath. So yeah, that way even the cell wants to conserve as much energy as possible. So for the formation of unnecessary products, it shouldn't be wasting its energy. So that's why it'll try and conserve the energy by switching off genes whose products are not required by the cell. Now, certain gene products may be required by cells at all times. They will be permanently on. For gene products which are not required all the time, they will be turned on and off based on requirement. So how do you regulate it? That is what we're going to learn right now. Regulation in eukaryotes can happen at 4 levels. See, for a gene to be expressed in eukaryotes, first it has to get transcribed, then post-transcriptional modifications have to happen, then it has to be transported out from the nucleus into the cytoplasm to the ribosome, and then translation has to happen. If you do not want a eukaryotic gene to express, you can stop the expression at multiple places. So either you stop it at the transcription level itself and make sure that transcription itself does not happen. If transcription doesn't happen, obviously gene expression will be stalled, right? Or let's say transcription has already happened. We will make sure that the post-transcriptional modifications don't take place. If hnRNA is not converted to mRNA, it does not become functional, it cannot get translated. Then it has already gotten processed. You will prevent the transport of that mRNA from the nucleus to the ribosome, right? That is one way you can stop expression. And then if it has already moved into the cytoplasm, it has been transported, then you find a way to stop translation from happening. At least that way you can conserve energy. The sooner you do it, like if you're stopping it at the transcription level itself, then you conserve more energy. Because for transcription, some energy will already be utilized, right? And that's wasteful. So the sooner you regulate it, the more energy the cell can conserve. However, in prokaryotes, whenever regulation of gene expression is happening, it mostly happens at the transcription level itself. There is one wonderful example for you to understand how regulation of gene expression happens in prokaryotes, and it's called the lac operon. Over here, lac means lactose, not lac rupees. LAC, lac, refers to lactose. Lactose is a disaccharide that contains glucose and galactose together connected by a glycosidic bond. And operon means what? Operon is a polycistronic structural gene. I already told you what is polycistronic. It means in one mRNA, there's information for more than one protein. So in this case, there is information for 3 proteins. So this is regulated by a common promoter and regulatory genes. There are these 3 proteins whose expression is being controlled. Okay, one polycistronic gene that is going to produce 3 proteins, and the expression of all these 3 proteins is controlled by one set of regulatory sequences. It's like one switch you have. One switch that you have controls the main light in your bedroom, the bed light in your room, and also the fan in your room. So if you turn on that, all the 3 will get turned on. If you turn that off, all the 3 will get turned off, something like that. So you have one structural gene that is responsible for the formation of these 3 proteins, and that structural gene has a common promoter, and these 3 genes have a common regulatory sequence which is acting like a switch. If this is on, all 3 will be produced. If this is off, none of those will be produced. Okay, something like that. So now let's understand the structure of the operon. These are the structural genes, these 3. Lac Z, lac Y, lac A. There is a promoter region. You know, in all genes, there is a promoter and a terminator. This is the promoter for these structural genes. So this is a promoter, and this is an operator sequence. This acts like the switch operator. Okay, so this is what acts like the switch. Now, apart from this, there is another regulator gene. Okay, so regulatory sequence. So, this is called the I gene. I gene has its own promoter. This regulates the expression of these genes. Okay. Now, this I gene produces an mRNA called repressor mRNA, and that eventually produces a protein called the repressor protein. The products of these three genes are beta galactosidase, permease and transacetylase. They are required for metabolism of lactose. Like, let's say this is something that's found in bacteria. So if you have a medium where bacteria are growing, or let's say it need not be a medium, artificial medium in the lab, wherever they are naturally growing also, in the medium where they are growing, if glucose is not present and lactose is available, if glucose is there along with lactose, they will want to use glucose itself, because that's the most preferred substrate in all organisms. But in the absence of glucose, if lactose is present in the substrate, they'll try and make use of that. They will take in lactose into their cells, they will break lactose down into glucose and galactose and then start using it for its energy requirements. Okay. For taking lactose present in the medium into the cell, you require an enzyme called permease. Oops. Yeah, permease. This is expressed by Y gene. Y gene is responsible for the production of this enzyme permease. Beta galactosidase is an enzyme that breaks down lactose into glucose and galactose. So, if this cell has to take up lactose from the surrounding medium, break it down and utilize it, these enzymes are required, and in the metabolism of lactose, transacetylase is also an enzyme that is essential. Okay, so it performs something called a transacetylation reaction. So, all these three enzymes are required for metabolism of lactose in bacteria. But if the medium does not have lactose at all, or along with lactose, if glucose is also present, they will prefer to use glucose. Okay. So, whenever no glucose is there, only lactose is there, then only you require these enzymes. Otherwise, you don't require them. So, how is that regulated? So, when there is no lactose in the medium or glucose is present along with the lactose, so these enzymes are not essential, this I gene produces this repressor protein. This repressor protein will go and happily sit on top of the operator. It will go bind to the operator. Okay. Now that this operator is present between the promoter and the structural genes, for these to get transcribed, RNA polymerase along with the sigma factor should go and bind here. It goes and binds there along with the sigma factor. But it cannot proceed any further because the road is blocked. If it cannot move and transcribe these, transcription will not happen, translation will not happen, enzymes will not get produced. Right? So that way it will turn off the structural genes. What happens if lactose is present in the medium? If lactose is present in the medium, this repressor will bind to the lactose. Inducer here is either lactose or allolactose. This repressor can either bind to this or it can bind to this. Earlier, it did not have lactose, it had only one option, it went and bound to the operator, prevented the transcription. Now that lactose is present in the medium, in the presence of lactose, it will bind to lactose. Once it binds to lactose, the repressor becomes inactivated, its shape will change such that this can no longer bind to this. If this can no longer bind to the operator, it's a free road for RNA polymerase. It will go bind to the promoter, transcribe, produce this lac mRNA, and ultimately it will get translated and these enzymes will get produced. These enzymes are involved in the metabolism of lactose and they obtain energy from that. Again, if the lactose is used up, there is no more lactose present, repressor will go and bind to the operator. Okay, so this way it will make sure that the genes are expressed only when the products of those genes are essential by the cell. In this case, we call lactose an inducer because that is what is inducing the expression of these genes. In the presence of lactose, the genes are turned on. In the absence of inducer or lactose, you don't need them, so it is turned off. That is how it gets regulated in bacteria. Okay. Now we've almost reached the end of the chapter. We have just two more concepts to finish. You've held on for this long. Just keep up for a little bit longer and we'll be finally done with this chapter. We are dealing with Human Genome Project right now. Following this, we will learn about the DNA fingerprinting technique and stuff, and then we'll be done with this chapter. Okay. So what is the Human Genome Project? This is one huge mega project that was, they started conducting this in the 1990s, very long ago. And this project is all about sequencing the human genome. You know what a human genome is? Genome is one full set of genetic information. So if you consider humans, humans have 22 pairs of autosomes, and we have two sex chromosomes, X and Y in males, and in females you have two X chromosomes. So in this project, their intention was to sequence the DNA in all the chromosomes in one set of genetic information. Okay, so it's not like they hadn't done it earlier. They had done sequencing of different regions of the genome, but the entire genome of humans to be sequenced, this was the first time. So what they did was they took 22 autosomes, one of each pair. We have 22 pairs of autosomes, so they took 22, one of each pair, and one X chromosome and one Y chromosome, totally 24 chromosomes, and they tried to figure out the sequence. I mean, A, T, G, C, C, whatever is the sequence of all of those 24 chromosomes, they tried to find out. Okay. So, this project was launched in the year 1990. It went on for 13 years until 2003. And this was coordinated by two organizations in the United States of America. One is the US Department of Energy and the other one is National Institute of Health, both from US. Along with that, there were many partners that collaborated and coordinated in this project. One is the Wellcome Trust from UK. Apart from that, Japan, China, France and Germany also partnered. Okay. So it took these many countries and that many years to sequence one person's, one cell's, one set of genetic information. In fact, it is called a mega project because of two reasons. Firstly, the amount of money this project cost was huge back in the 90s, and also the amount of data that was generated because of this project was huge. So it was called a mega project. First, let's talk about how much money this project cost. You know how in the human genome, we learnt about it in the beginning of the chapter. In the first slide itself, I told you that human genome has 3.3 into 10 to the power of 9 base pairs, right? So we already know that many base pairs are present. So if you roughly take it as 3 into 10 to the power of 9 base pairs, back in the day in the 1990s, for the sequencing of one base pair, it cost $3 and we had 3 into 10 to the power of 9 base pairs to sequence. So if you multiply that, the cost of this whole project was $9 billion in 1990s. Okay, so if we do not understand billions and millions well, so if I have to put it in terms of how much it cost in crores, it cost 900 crores, not rupees, 900 crore dollars. Okay, so that's how much money this project cost. And talking about how much data it generated, 3 into 10 to the power of 9 base pairs is a very, 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 very long stretch of DNA. So if you were to store this information in the form of books, if you were to print them in the form of books, assuming you will have 1,000 letters, each letter representing one nucleotide in one page. If you have 1,000 letters in one page, 1,000 such pages in one book, we would need 3,300 such books to store the information. So the information that we would obtain as a result of this project was so high that you couldn't like store them in physical copies. Why is this whole project being done? That is because you want to analyze different segments of DNA. You want to find out the difference between one gene sequence and another gene sequence. You want to find out what kind of proteins you can get. So you want to study these sequences, right? If you had like 3,300 books spread across, how would you analyze different sequences? That would become too difficult. So this necessitated the need for computer programs and databases where such a huge information could be stored and even analyzed. Okay, so at around this time, in the early 1990s to 2000s, there was a brand new branch of biology that kind of popped in, which was called bioinformatics. Using bioinformatics, you used basically computer programs to analyze DNA sequences, compare DNA sequences between two different organisms and all of that. So yeah, that kind of gained more popularity around that time. Now, what are some of the goals of the Human Genome Project? I already mentioned they wanted to identify every single base pair in the entire stretch of the human genome. So that was 3 billion base pairs. And of course, throughout the length of DNA, you do not have genetic information. You have it only in certain regions. Wherever genes are present, they wanted to identify the approximately 2000 to 2500 genes that are located in the human DNA. And whatever information we obtained, they wanted to store it in databases in order to analyze and compare the data. Whatever existing analytical tools were there, they were not sufficient. They had to improve those tools. And if there is any technology that is developed in order for this project to get completed, if that technology is used is beneficial in any other field, like drug research or agriculture or whichever field such technologies can be used, then yeah, transfer such technologies to other industries. Then if at all there is any ethical, legal or social issues that arise from this project, then address that. Okay, now let's talk about the methodologies that were used in DNA fingerprinting. There are two approaches that they took. It's not like for some segments they took one approach and for the other segments they took another approach. Both these approaches were taken hand in hand throughout the length of the human genome. First, we'll talk about sequence annotation, then I'll tell you about express sequence tags. Sequence annotation was done throughout the length of DNA. It's more like NCERT mentions that they took a blind approach to sequence the DNA here. So basically, regardless of whether that segment of DNA had information for protein synthesis or it did not, regardless of it, they went on and sequenced the entire genome. How did they do that? So they did that. Okay, first you take a human cell and then from that cell you isolate one set of genetic information, the chromosomes, and then you cut it. Okay, so what they did was, once you get this DNA, isolate the DNA from any human cell, one set of genetic information you have, you cut it using restriction endonucleases. Okay, there are many ways in which you can cut it. So you cut it into fragments because the length of DNA is so long that you can't sequence it as one stretch and you want more copies of it. So what do? After you cut the fragments, you clone it using certain host organisms like bacteria and yeast. You may have learnt in biotech chapters that you can't just take a random segment of DNA, fragment of DNA and force it into a cell and expect it to multiply. In order for a DNA fragment to make multiple copies of itself, it should have an origin of replication. It's not like our DNA everywhere we've cut it, there will be an origin of replication, right? So you need vectors in that case. So vector will have an origin of replication. You integrate your gene into the vector DNA so that every time the vector makes a copy of itself, it along with its DNA, it will make a copy of our DNA also. So the kind of vectors that they used in this case were bacterial artificial chromosomes and yeast artificial chromosomes. They are the vectors. Using bacterial artificial chromosomes, they introduced our genes into bacteria, and yeast artificial chromosomes were used as vectors to introduce our genes into yeast. Both of these are host cells. They multiply at a very rapid rate. Yeast cells multiply once in every 90 minutes and bacterial cells multiply they divide once in every 20 minutes. So every time it's dividing, it's going to make extra copy of our DNA as well. So you get more and more copies. Then what do you do with those copies? There is a sequencing technique or a method called Sanger sequencing. So using the Sanger sequencing, they could kind of, get to know what is the exact sequence of DNA present in that particular um, fragment. Okay. So Sanger sequencing is like a computer program that was developed by a person called Sanger. That's why it's called Sanger sequencing. So what it does is basically, I think from what I know, it is based on a fluorescence dyeing technique. You add certain dyes like chemical dyes, fluorescent dyes to the DNA molecules, and it passes through a very tiny pore, and every time it passes through a pore, it kind of emits a particular wavelength of light. And based on the wavelength of light, the computer program can get to know whether it is A, G, C, and T. And based on that, it will give you a sequence. It's not like you're looking at the microscope and there is A, T, G, C written on DNA and you will know, right? Obviously, we have given it names and according to that, we've given it like abbreviations ATGC. It's not like ATGC sitting on top of a DNA molecule. So making use of these fluorescent dye techniques, if it is appearing red, that is one nucleotide. If it's appearing green in color, that's another nucleotide. That's one way they figure out the sequence of DNA. We'll not get into the details of that. So that is how you will get to know the sequence of DNA. It's like this is where the DNA sample is introduced and here the fluorescent dye is added to the DNA. and based on what wavelength of light is emitted, the computer will tell you what is the sequence of DNA in the fragment that we have used as a sample here. That way you will get samples of many fragments. We've taken our entire DNA and cut it into so many fragments, right? Some of these fragments will overlap, okay? So based on the overlapping sequences, you kind of get to know what is the complete stretch of nucleotide sequence in our DNA. So that is what we do in sequence annotation. Sequence annotation. In expressed sequence tags, you specifically look at only those regions where information is present. That's why it's called expressed sequence tags. Okay, so ESTs they are called. If in a particular region there is genetic information, how will we know? Basically, you take the cell and you take its mRNAs. You take mRNA, you reverse transcribe it, get DNA, find out its sequence. After finding out its sequence, you would have had the entire DNA sequence using sequence annotation. Sequence annotation, right? So based on where the genes are present, these genes are present on the entire stretch of DNA, you can assign it locations and tell, okay, at this position, this gene is present. At this position, this gene is present. You can annotate that. So that is how the DNA project, Human Genome Project got completed eventually in 13 years. This is what they did. And of course, it's very time consuming. So it took a lot of time. Now, speaking of the human genome, after the Human Genome Project was completed, we got to know these are the features of the human genome. It contains 3,164.7 million base pairs, roughly 3 billion base pairs. There are many genes present in our human genome. Earlier, some people had estimated that it could be 80,000 to 1,20,000 genes and all of that. But after this experiment, we got to know that the number of genes in our genome is 30,000. Among those 30,000 genes, different genes have different lengths. So if you take an average of all the 30,000 genes, the rough average or length of a particular gene is about 3,000 bases. The largest known gene in the human genome is the dystrophin gene. It has 2.4 million bases. In two individuals, almost 99.9% of DNA is the same. The difference lies only in 0.1%. Of these 30,000 genes that were identified, we don't yet know the function of more than 50% genes. Okay, so it's yet to be discovered. You all can do that job. Yeah. Within the entire genome, it's only about 2% that codes for proteins. Less than 2%, not even 2%, less than 2% that codes for proteins. There are a lot of repeated sequences found in our DNA. They are called repetitive sequences. Sometimes they repeat so many times that it could range between 100 to 1000 times. Okay, so that's something that they got to know. Chromosome 1 has the most number of genes, which is 2,968. Chromosome Y has the least number of genes, 231. There are 1.4 million locations in the genome where there is difference between two different individuals in just one base pair or one base, you can say. They are called single base differences or single nucleotide polymorphisms, SNPs, and it's pronounced as SNPs. Okay, so these are some things that we got to know about the human genome after the Human Genome Project. So this of course can be used in a lot of ways. If there is any genetic disorder, then if you can personalize drugs for that particular genetic disorder based on the sequence and all of that, you can figure out using this information from the Human Genome Project. Now, apart from the humans, there are many organisms whose genomes have been sequenced. Examples: bacteria in the year 1995, yeast in 1996, Caenorhabditis elegans in 1998, Drosophila and Arabidopsis in 2000, and rice plant was also sequenced. The genome of the rice plant was also sequenced in the year 2002. With that, we have come to the end of Human Genome Project. We have a last concept to finish, which is DNA fingerprinting. I hope you've kept up until now. We'll talk about DNA fingerprinting. This technique was, devised kind of by like a person by name Alec Jeffries. We already learnt here two things. in our DNA, there's a lot of repetitive sequences, one. Secondly, 99.9% of DNA in all individuals is the same, right? So we will use this in DNA fingerprinting. DNA fingerprinting has nothing to do with this fingerprinting. DNA fingerprinting is also unique like our fingerprints. That's why it's called DNA fingerprinting. Basically, if you want to find out the identity of a person, of a criminal, or if you want to do paternity testing and find out who's the father of a child, all of that, you can't expect to take the DNA of these two individuals and compare it, sequence it, because you know how long it takes to sequence DNA. It's very time consuming, money consuming, effort consuming, and all of that, right? So, if there is an easier way to find out about the identity of a person using their biological samples, then one such way is DNA fingerprinting. This is done routinely nowadays in order to find out who the criminal is, who is the father of the child, for paternity testing and all of that, forensics and all of that. DNA fingerprinting is routinely done. If you watched crime series like Dexter or Crime Patrol, you will frequently hear people talking about DNA fingerprinting, right? So yeah, that's that. Since it is impossible, not impossible, I would say, it is not feasible to find out about the identity of a person by sequencing their entire DNA. What we do here is much simpler. We know that the human genome has how many base pairs? It has 3 into 10 to the power of 9 base pairs and one set of genetic information. We know that 99.9% of it is the same in all individuals, so the difference is only in 0.1%. We are only going to focus on where the differences are. So 0.1% of 3 into 10 to the power of 9 base pairs is 3 into 10 to the power of 6 base pairs. It's not like we're sequencing this. See, this is also a huge amount of DNA. To sequence this also will require a lot of money. We're not going to sequence this, but we are going to look at the repetitive sequences found in this region. Okay, so there's a lot of repetitive sequences. Usually they are found in non-coding regions. That is our prime focus for DNA fingerprinting. That is where the principle of DNA fingerprinting lies. Okay, so what exactly are we going to do in DNA fingerprinting? I'll tell you. Firstly, if you take our DNA, in our DNA, you have bulk DNA and satellite DNA. Now, what is bulk DNA? What is satellite DNA? I'll tell you. When you spin our DNA in what is called a ultracentrifugation, cesium chloride density gradient ultracentrifugation, we spoke about it in the Messelson. Oh my god, what is this, stammering so much from yapping since I don't know how many hours. Yeah, so we studied about this in Messelson and Stahl's experiment. It's the same thing. You isolate DNA from any biological sample and spin it in a centrifuge. You will get bands. Okay, so you will get some thick bands and some thin, smaller bands. The thicker bands that represents the genomic DNA, and the smaller bands are the satellite DNA, we call it. So when you take this, when you isolate the bulk genomic DNA and satellite DNA, and you pass it through, pass light through it in a spectrophotometer, then you will get to know the absorbance. If you plot the absorbance on a graph, then you're going to get peaks like this. NCERT has just mentioned you get a big peak, small peak, and all of that. So basically, this is the large peak, this is the smaller peak. Many small peaks like these are called satellite DNA, and they will have a lot of repetitive sequence, and we are going to utilize only the satellite DNA for our DNA fingerprinting. Okay, so there are different types of satellites you can find based on whether it has more AT, more GC, how long is the repetitive unit, is it short, small number of times it's repeated, or is it repeated multiple number of times. Based on all of that, you can classify satellites into many types. Now you will classify satellites based on the length of the repeating unit and the number of repeats. You have 3 different types. You have microsatellite, mini satellite, macro satellite. Microsatellite has the repeating unit that is 1 to 6 base pairs in length. So ATT, GCCA, like that is one sequence. The same thing will keep on repeating many times. Okay. So the length of the repeating sequence is 1 to 6 nucleotides or base pairs, and that can be repeated as many as 100 times, tens to hundreds of times it can be repeated. Short tandem repeats is one type of microsatellite. We'll talk about mini satellites. Mini satellites are slightly longer in the repeat unit, and the number of times it gets repeated is also much higher. So this can range between 10 to 60 base pairs, and it repeats from 100 to a few thousand times. One example for that is VNTR. This is what we're going to use in DNA fingerprinting, a type of mini satellite called VNTR. VNTR is short for variable number of tandem repeats. Then we have macro satellites, which are not important. The repeat sequence itself will be greater than 100 base pairs. Okay. So what are these VNTRs? They are variable number of tandem repeats. For example, if this is a repeat sequence, on either sides of it, there will be restriction sites. Okay. Sites for restriction endonucleases. Here, the sequence could be A, G, G, 1, 2, 3, 4, 5, 6, 10. Okay. So this is the repeat unit, let's say. How many times this unit is repeated in an individual varies. This sequence is the same. How many times it gets repeated varies between 100 to thousands of times, right? In me, it could be 300 times. In you, it could be 470 times. In another person, it could be 999 times. So, how many times this gets repeated is variable from one person to another. If it's gotten repeated many number of times, then the length of this segment is going to be more. If it's gotten repeated fewer times, then the length of this fragment of DNA is going to be much lesser. So that's why we call it variable number of tandem repeat. Tandem repeat means what? This should continuously keep getting repeated one after the other with no other sequence intervening in between. That's called a tandem repeat. And the number of times it gets repeated in one person and another person is variable. That is why we call it a variable number of tandem repeats. Okay. So this is the basis for DNA fingerprinting. What we do is we take first from a biological sample. Wait, before that, I'll tell you how these variations happen in different peoples. It happens because of mutations. You know, in all of us, mutations happen all the time. Mutations can happen in our body cells, somatic cells, or it can happen in the germ cells. If it happens in the somatic cells, there is no way it can get passed on to the next generation. If it happens in the germ cells, it can happen in the coding sequences, it can happen in the non-coding sequences. If it happens in the non-coding sequence, obviously there is no bad consequence of it. So it will obviously get passed on to the next generation. If it happens in the coding sequence, the mutation could be detrimental to that individual, and that individual could have impaired reproductive ability. So he or she is no longer able to bear children. So that in that case, it will not get passed on from one generation to next. If it has happened in the coding sequence and it has not impaired the reproductive ability of that person, then it will get passed on to the next generation. So like this, because of mutations happening in us, in germ cells, in the coding sequences where reproduction is not impaired, or in non-coding sequences, such differences, changes in DNA will keep on getting accumulated in population. So basically this happens because of mutation. There is a mention of DNA polymorphism in your textbook. What is DNA polymorphism? It is a type of mutation where that particular sequence, okay, that should be present in 0.01% of the population, then you can call it a DNA polymorphism. Or to put it in words that you can understand, if there's a population of 100 people, one among 100 people, if that particular sequence exists, then it is equivalent to a DNA polymorphism. Among 1,000 people, if 10 people have that sequence, then it is called a DNA polymorphism. If less than 10 people among 1,000 have that, then you cannot call it a DNA polymorphism because it is just called a mutation. Okay, for it to be called a DNA polymorphism, at least 0.01% of the population should have that sequence. Okay, so we're basically working with those sequences only in DNA fingerprinting. Now, coming to the steps of DNA fingerprinting, first you need the biological sample to isolate the DNA. The biological sample could be a hair, could be your skin shreds, could be your blood, could be the semen, could be the saliva, it could be whatever. Okay, so you need one biological sample. From that, first we will isolate the DNA. You know how to isolate the DNA and all of that. You've learnt about it in the biotech chapter. And then what we do after you've isolated the DNA is you will cut it into short fragments, pieces using restriction endonucleases. After that, you will separate the fragments based on I was about to sneeze, but somehow it's over. Yeah. Based on the length of the fragment, the DNA mixture will get separated in a gel electrophoresis. About gel electrophoresis, you would have learnt in biotechnology chapter. So it's basically separating the DNA fragments based on its size. The smaller ones will move quicker towards the anode and the larger ones will stay behind. So a banding pattern will form on the gel, agarose gel, when gel electrophoresis is performed. Of course, this is required. This itself will give us like the DNA fingerprint, but this is not stable. That's why you will need some stable copy of the DNA fingerprint. So what do you do next? You will take this gel and place it in an alkaline solution and make it single-stranded. The hydrogen bonds between the two DNA strands will break and the DNA will become single-stranded. After it has become single-stranded, you will transfer the DNA in the gel to another paper. Okay, so that could be a nitrocellulose paper or a nylon membrane. That process is called southern blotting. So because of capillary action, if this is the gel, this is the paper, and you keep a little bit of weight or paper towels or something on top of it, because of capillary action, it will get transferred to the nitrocellulose paper. Once it has gotten transferred to the nitrocellulose paper, you will add VNTRs, radioactive probes. Okay, so VNTR probes we call them. So you only have cut the DNA into fragments. You know which VNTRs you're looking at. So you would have artificially in the lab synthesized these sequences complementary to this, whatever is there. Those sequences you would have artificially synthesized in the lab and made it radioactive. Okay, so that you will add along with this nylon membrane, nitrocellulose paper, whatever, and rock it for a little bit, and then wherever complementarity is there, that will go and bind. Binding of VNTR probes along with the single-stranded DNA is called hybridization. Following hybridization, you will keep this nitrocellulose paper against an X-ray film, because wherever radioactivity is there, over there bands will form on the X-ray film. That is called autoradiography. Ultimately, what you get here is your DNA fingerprint. So you can see in the first step, isolation of DNA will happen. Second step, restriction endonucleases are used to cut it. Third step, using gel electrophoresis, based on its size, you will separate it. Obviously, in one person, the number of repeats will be long. Their DNA fragment will be longer. In me, the number of repeats is less. My DNA fragment will be shorter. So like that, from between two individuals, there will be differences in the length of DNA. That's how you get different banding patterns. So after this, they will make it single-stranded. That step is not mentioned in NCERT, and then they will transfer it to a nylon membrane. The process is called southern blotting. Following that, they will use VNTR probes to hybridize the single-stranded DNA and then place it against an X-ray film to get the pattern on top of the X-ray. That is called autoradiography. So once they do this, they will get the final DNA fingerprint. Here is an example of how DNA fingerprinting, very oversimplified example this is, where they've taken DNA from two different individuals. It's possible that these two are the suspects of a crime. At the crime scene, they would have obtained one sample. They are calling it C. Okay. And in the crime scene, at the time when the crime happened, there were two people probably, and they've taken the biological samples of those people also. And they've done a DNA fingerprint. They've run a DNA fingerprint from this sample, from this person, from this person. And if you look at this, if you compare the two, you can see how this person's, banding pattern and this person's banding pattern are identical. So this kind of tells that this guy was at the crime scene when the crime happened. So he's probably involved in some way. Yeah, so you can tell that. This is very different from this. So you can't say that this guy, this biological sample at the crime scene does not belong to this guy. That's what you can say. Okay, so with that, we have come to the end of this chapter, Molecular Basis of Inheritance. Mighty big chapter. I've been yapping since the last, I don't know how many hours. I hope this was worth it. I hope you've understood all of the concepts. For the amount of talking that I've done today, my jaw has gotten so much exercise that I should have developed a jawline that should cut through apples right now. But nothing has happened. It's okay. I hope this session was helpful. If you have any doubts or anything, you can let me know in the chat and I will answer them. And yeah, I'll see you again in the next class. Please keep studying hard. Exams are coming very close. They will approach you so much sooner than you realize. So just keep hustling hard. Yeah, all the very best. Thank you.